आउटपुट हाँ ओके हेलो हेलो सर के पास देखो ना, मैं अभी हेलो 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 सर को आवाज नहीं जा रहा है सर। ये डेस्कटॉप ऑडियो ऑन करना पड़ेगा ना मेरा। Send on issue of pharmacy and research. सर इनपुट किधर लेट आएगा ना यूट्यूब पे। हेलो हेलो हाँ आ रहा है सर के पास देखूंगा मैं अभी। हेलो 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 इनपुट कैप्चर चलो। ये डेस्कटॉप ऑडियो ऑन करना पड़ेगा ना मेरा।
थोड़ा लेट कर रहा हूँ।
हाँ लेट हाँ सर ओके
हाँ लेट हो 1 मिनट का लिया तुमको।
चल तर हे गुगल मीट मध्ये कोड हे करायला पाहिजे होतं।
काय करतो
गुगल मीट मधून पण
आता शाळा झाली।
हेलो हेलो हाँ हाँ सर मैं मीट इधर ये मीट चालू करूँ क्या, मीट शेड्यूल करूँ क्या तो फिर हाँ हाँ चलिए सर निशा मैम क्या बोलू ना ये इसमें गूगल मीट भी ये करो ना, मीट का भी ये करो शेड्यूल करो, मीट का भी शेड्यूल करो,
मीट का भी शेड्यूल करो,
दोनों लिंक करो, मीट का भी कोड ये करो।
आप कैलेंडर में शेड्यूल कर रहे हो ना।
हाँ
तो ए डिवीजन एंड बी डिवीजन अलग अलग शेड्यूल करना।
लेकिन फिर कैसा रे।
ये भी लिंक भी रहने दो।
हाँ।
और अपना गूगल मीट का भी ये चालू करो,
सेव करो अभी।
हाँ
अभी दूसरे डिवीजन में भी सेम करो, दूसरे डिवीजन में भी सेव करो। 1 मिनट हाँ
मतलब एक ही मीटिंग सबको ज्वाइन करने के लिए बोलो, डिवीजन 1 डिवीजन 2 को
सेम
एक से ही सब ज्वाइन करें।
लेकिन 100 ही होगा ना।
हाँ चलेगा ना, जिसको नहीं ज्वाइन हो उसको बोलने का लिंक में से यूट्यूब लिंक से ज्वाइन करो।
अभी मैंने दोनों डिवीजन में एक ही मीटिंग कोड डाला तो एक ही में जाएंगे दोनों।
हाँ एक ही
पक्का ना, क्योंकि देख दोनों में एक साथ नहीं शेड्यूल कर सकते।
क्योंकि इसमें आवाज है ना, इसमें आवाज है ना 30 सेकंड बाद जाता है। हाँ यूट्यूब
देख अभी इसका है ज्वाइन विथ सेम लिंक है TZQ। और ये
गूगल मीट करेंगे ना
हाँ।
जब भी चलेगा तो इनको सर को भी दिखेगा वो।
हाँ
हाँ और बच्चों लोग को भी दिखेगा।
ये देख दूसरे डिवीजन में भी सेम लिंक है तो दोनों एक ही पे जाएगी। कोशिश करनी। चल मैंने अभी B से ज्वाइन किया। अच्छा A से ज्वाइन करके क्या आ जाता है। हाँ तो फिर वो मीता मैम को मैं ये कोड भेज देती हूँ, वो भी ज्वाइन कर लेंगे, वो आए नहीं है अब तक। नहीं आएंगे आई मीन
उनको तो दिखेगा ही ना
हाँ।
आपके इसमें।
मित्र मैडम नहीं आने वाली है ना?
नो हाँ सो आई विल कोऑर्डिनेट विद देख ज्वाइन किया
सर आपल्या तुमच्या
ए में
व्हिडिओमध्ये
ज्वाइन
काय
ए में
दाखवलं?
ज्वाइन
सर
एक ही आहे.
तुमच्या व्हिडिओमध्ये बघा काय दाखवलं.
ए में ग्रीन वाला बस हाँ सेम ए से ज्वाइन किया ना ये मैंने एम एक में जा रहा हूँ बराबर
ये इतना आवाज लेट आ रहा है ये कैंसिल किया.
नहीं नहीं अभी ज्वाइन नहीं करनी है ना मैं खोल के रख देती हूँ यहाँ से ज्वाइन कर लेंगे जिनको ज्वाइन करना है जाना और हर्षद एक 2020 वाला टेस्टिंग वाला ना अभी भी चैनल में पड़ा है सुबह मैंने देखा डिलीट करके हाँ
ये क्या है ये कौन प्ले करेगा.
क्या ये है क्या पावर ऑफ ड्रीम रहेगा ना अपना?
पॉवर ऑफ ये सब
अपना प्रेयर है क्या इसके अंदर प्रेयर और पावर ऑफ ड्रीम
हाँ है ना.
खोल के रखना फोल्डर यही रहेंगे ना तुम में से कोई एक देख वो सब वो फोल्डर खोल के रख दे और मैं बाकी सबकी पीपीटी डाउनलोड ये है कौन सा वाला है सरस्वती वंदना नहीं चाहिए अपने को ये अपना सेंट फ्रांसिस प्रेयर है ना और कॉलेज का वीडियो है
कॉलेज का ही कैंपस.
अच्छा वो दोनों है ठीक है तब तो मैम ये पावर ऑफ ड्रीम अभी का है
हाँ अभी का.
तो ये दोनों प्ले करनी है मेरे को याद दिला देना अगर मैं बोलूँ ठीक है ये सबके डाउनलोड करके रख देती हूँ
Madam, I want to show it from pen drive. Then how it should be done?
you just have to plug in
हाँ.
see I have opened the Google Meet you just have to join the Google Meet and I will keep be joined otherwise हम लोग बाहर हो जाएंगे अगर 100 participants हो जाए तो then you just plug it in as usual
Then screen sharing and all that has to be done.
हाँ see just present now we have to
हाँ.
present now then you present the screen which is there
So whoever is sitting here, they have to click to the link.
no whoever is sitting here directly plug in and play
Acha, the share screen and everything will be on only.
on I just talked to madam
सेकंड है 2 मिनिटात तर आवाज
तर ते ह्याचं पण असतं ना इंटरनेट स्पीडचं पण असतं ना काय होतंय ते मेन गोष्ट आहे की लाईव्ह स्ट्रीमिंग आता 400 जण पण बघू शकतात ते
गूगल मीट मध्ये 100 आहे गुगल 5 आहे पूर्ण फॅकल्टी ते कसं आहे
मग वरती युट्यूब वरती
फॅकल्टी युट्यूबवर म्हणजे
फॅकल्टीने आता जॉईन करायला पाहिजे
मैंने ज्वाइन करके रखा है
हा फॅकल्टीने आतापासून जॉईन करून ठेवलं पाहिजे म्हणजे जे बाकीचे पोरं येतील ना ते युट्यूब वरती जातील
105 दिसतोय
हाँ सर इट इज ओके बिकॉज मैंने पहले ही मेल किया है जो प्रेजेंट कर रहा है वो यहाँ से ज्वाइन करे जो नहीं प्रेजेंट कर रहा है
इन केस 100 बच्चे ज्वाइन कर देते हैं तो फैकल्टी को
यूट्यूब पे देखो
हाँ यूट्यूब पे लेकिन जिसको प्रेजेंट करना है
वो यहीं से करना ऑलरेडी इसलिए मैंने पहले ज्वाइन भी तब आपको मैं बोल देंगी एक मिनट
सी somebody has joined
हाँ
वो बंद होकर देना पड़ता है आपने वो मैसेज किया ना, लिंक जॉइंट लिंक भी कर देंगे बच्चों लोग को।
Good morning. Prayer is there.
Yeah.
So that only I came to tell you that we have like.
I have seen this. No problem. So I'll wait here. No. But where is the camera for this? The laptop directly.
Yes.
Laptop camera only.
See,
So where to look exactly?
Here, I have already kept joined, so we don't have any problem of not joining after the students have joined.
Huh.
So we will record from here only, and on the YouTube it will be like this visible
Okay.
after 10 o'clock. Now it's not streaming. We just switch on.
So you just want only 10 minutes, na? Just introduction, because remaining all points I don't need to cover.
Yes.
So I don't have to repeat everything. I mean, I don't have to say much. I will just say welcome, and then I will talk to them, and I will just. I'm not preparing any slide or prepared anything. It's just going to be a short welcome address.
So while you speak, you want me to put up a introduction slide like that induction one template?
Let them see me, they'll recognize at least when they come here.
They are very chunt.
Huh?
They are very chunt. This time students are like very smart. I and Varsha had to give them a form.
But all have joined now, all 105?
Yes. Now yesterday we circulated the roll number list also as final admission is done. So they are very well aware of which division and all. Now they are to be accommodated with the departments and MIS. So I have already coordinated with Mita ma'am. She will be presenting from there.
Huh, Meeta is not coming today.
Yeah, she said that. हर्षद, ये सबके पीपीटी डाउनलोड करके रखो कि मेरे मेल में पड़े।
डाउनलोड करके रख दो ना।
अगर वो प्ले नहीं कर पाए तो
वो फोल्डर रखा है ना, देख सकते।
हाँ, फोल्डर मीता का MIS फोल्डर है, उसमें डाल दो।
नाम दे दो ना।
इसमें पड़ेगी क्या? Of your beats. क्या किया भाई? मैं कर रही हूँ कुछ।
प्रेयर रखा है, प्रे करना है।
हाँ, मैंने बोला हर्षद ने ये सागर ने दिखाया मेरे को काम। ओके।
How will you know that they are attending?
We will see the number in YouTube also, and we will see the names in here also.
We'll get the names in this. This you will get 99.
Huh. There we can see who has joined, number of people who have joined.
And ask them in the YouTube chat. They should put their name and roll number.
Okay. I'll ask them.
चार्ट बंद करेंगे
चैट बंद किया ऑन कर देना डाउट रहेगा तो फिर 1 मिनट रुका ये कॉपी करने की 1 मिनट
YouTube chat असतं ना? That is off. Keep it on. No? They have to put their attendance on every lecture. Tell them in every session.
हाँ
When every session starts, those who are attending through YouTube should put their name and roll number. New roll number as per the division. Hello. Yes, Sister. Good morning. Yeah, we are having our student induction now, so I have to address. So maybe after 10:30 only. By 10:30 I'll be free. But after 10:30. Huh? Yeah.
वो लेट नॉइस के लिए
डिलीट किया ना तुमने वो नहीं सुबह मैंने देखा तो 2020 था
हाँ नहीं वो है अभी भी वो बाद में डिलीट कर देते
हाँ अभी कुछ मत कर मैं कुछ कर रही हूँ
गुड मॉर्निंग का ईमेल पे देखो चार्ट मिलता है कभी कभी यूट्यूब पर
कहाँ डालूँ हाँ तुम हाँ तो डाल दो हाँ डाल हाँ आया ना हाँ ओके ठीक है
So I will also tell them that universal human magnet is anybody going to address it. So there is that I have forwarded it to Norma Mani. Do you see that? She showed me. There is that link which is to be followed. They should listen to those videos and there will be that by the end of the day you all can inform them.
हाँ मैम
By the end of the day you all can inform them. I'll give you by the 10 exactly, but I'll make a mention of it.
ओके मैंने सबकी पीडीएफ डाउनलोड करके व्हाट्सएप पर डाला
क्या हुआ
इन्होंने क्या भेजा है शेयर्ड इन ड्राइव भेजा है
Who is talking about academic calendar? Calendar.
academics I'm going to play this also, no? St. John Institute of Pharmacy and Research is the only college in Mumbai University who has received accreditation both for NBA and NAC. The success of these accreditations is all under the face that have helped the college to escalate to such a level. Let us now have a glimpse about the journey of St. John Technical and Education Campus. Let's go. Now I'll request Dr. Savita Tauro, Principal of St. John College of Pharmacy, to speak a few words to you all. Good morning, students. Am I audible to all of you?
Good morning, Miss.
Good morning and welcome to all of you to St. John Institute of Pharmacy and Research. This is not a normal situation where, in regular times we would have had you in the college campus and we would be addressing you in person. Unfortunately, it's an unprecedented time where we are connecting with you virtually, but nevertheless, it is very essential that we had to have this induction program for you so that you are made aware of the various aspects that you would need to know as if you were in the college campus. So congratulations to all of you for having secured the admission to the program of pharmacy. And I'm sure that for most of you, this was the career that you were looking out for. Some of you may have been aspiring to get into medicine or, some of the other medical professional fields. And probably you may be here not really out of your choice, but probably because you had no other choice. But since you have decided to take up this profession, let me tell you that this is probably one of the best decisions that you have made. And within a short span of time, that's within 3.5 years, I should say, you would be a graduate and will definitely be contributing to the field of medicine, maybe not directly with patient care, but surely working for the well-being of humanity. So what would you expect when you come into this program? If I look at the academic calendar as such, I'm sure all of you, including your parents, probably some of the parents are also watching this along with their wards. And some of you may be wondering how this program will go on since you are commenced the academic year 6 months later than the normal times. In a regular time, we would have commenced college in August. You have started college in February. You've lost 6 months of academic time. But the college, the faculty, the university have all planned it well so that you will still graduate within the stipulated time of 4 years. So you've joined in 24, that's in June 24. You should be completing your course well within time, and all the effects of the pandemic will no longer have affected your academic career. But for that, we will need to be doing a little bit of extra work during the first year so that we are able to complete the course and make up the pace so that we can catch up with the second, third, and the final year and be well in time to appear for final examinations. And I'm sure some of you may be aspiring for higher studies, for going abroad, and so on. And definitely, we would love to have our students fulfill all their dreams. So let me just give you a brief about the academic calendar as it would progress for this year. You have just commenced in February. You started classes on the 1st of February. And this semester, you will follow a semester pattern. You will have 8 semesters. The first year comprises of semester 1 and semester 2. And semester 1 will be taken up till the end of April, including the end semester examinations. The detailed academic calendar will be presented to you later and will be also shared with you. And so your first semester will be completed by end of April. You will have a break of probably just a week, and then we will commence the second semester in May. So second semester will commence in May and will go on till mid-August. So the plan for this academic year is from February 1st to 14th of August, you will be completing your first year B.Pharm. Now you may be wondering that we have to complete everything within a span of 7 months. Yes, so what are we going to cut short? Not the academics, but surely some part of maybe your
the break period that you would have, the gap between the 2 semesters, and yes, we will have to take up some extra lectures, some extra practicals, because you don't want to lose out anything on the technical aspect of your study. While we do understand that it may be a little hectic for you and you have already gone through a long period of waiting, probably a lot of stress, waiting for when admissions would happen and what would be like how things would go on. But I think it's time for all of us to put all of that at the back of our minds, look forward to a good beginning, ensure that we are able to make up and catch up with the rest, and see that we are in time for what we aspire to be in 4 years. So you will have to, as I mentioned, work a little bit extra. Faculty, your class in charges and the faculty will give you the schedule for how we will be having maybe some extra lectures and practicals. You'll wonder how this course has got a lot of practical work. So in normal times, you would be spending on an average at least around 16 hours in a week in the laboratory. So how are we going to do that? Fortunately for us, university has announced, government of Maharashtra has also announced that from 15th of February, we can call students to college, so that there is only about 50% of the student strength in college. There was a survey form sent out to all of you, and I'm sure your parents, you've also informed your parents. We are waiting for further guidelines from the university, but we would want students to come to the college because we will conduct practicals in college, and probably you would have to come only for 2 days in a week. We are not expecting that you should be here for the entire week. 2 days in a week when you can complete all your practical courses. Rest of the days you would have the theory courses online because that can be done online, but practicals, you would only be able to show you demonstrations and you would never get a hands-on experience of how things are to be done in the laboratory. So prepare yourself mentally for this part of the academic work, where you will have to start coming back to college, and of course that experience is going to be totally different. I know that you've been some of you probably had joined college for joined BSc programs or some other programs, so have already gone through half a semester, and it has been through online mode. And what we've got to understand is many students are now beginning to feel very comfortable with the online mode. But that would be okay if it was a very theoretical course. Yours is a practical curriculum, and therefore coming back to college to work in the laboratory is going to be very essential for you. Now, while all this, the details of all the academic program, the faculty team will be talking to you about different aspects during the day. So what I would like to mention to all of you, dear students, while you are here in this college, in this campus, definitely improvement in your technical skills, improvement in laboratory skills, gaining new knowledge, all of that is very, very essential. You are in a very competitive world. Remember that when you graduate, there are probably, if you've been looking at statistics, looking at what is there in the newspapers, if you just read newspapers yesterday, there are around 25,000 plus seats for Bachelor of Pharmacy in only the state of Maharashtra, which means these many number of students are expected to graduate along with you after 4 years. They have all taken admission. They will all graduate along with you. And all of them will be competing for whether it's post-graduation, whether it is a job opportunity in the industry, whether it's a post-graduation abroad or an MBA program. So all of these are your competitors, and the number of students who are competing each day for the higher studies, for placements, is all increasing. And all of them will be completing the same curriculum. Today we are following the curriculum which has been structured and which has been prepared by the Pharmacy Council of India. And now this curriculum is also universal. I wouldn't say universal, but it is uniform across the country. So all colleges in the country are following the same syllabus. So there is the curriculum part of it is what everybody will be doing. But what is it that you are going to gain additionally to make yourself maybe a candidate of choice for the next step? See, your first step into getting into B.Pharmacy, getting into the pharmacy program is already done. And you are one among those 25,000 odd students in the state of Maharashtra. You're probably one among
maybe over a lakh, maybe 2 or 3 lakh students in the country of India. So if you are looking at the next step of your career progress in this profession, you need to be doing something beyond just the pharmacy curriculum. And that will include getting into maybe some additional certificate programs, doing some kind of additional extracurricular activities, and involving yourself in all that the college will provide you. We are aware and we want to ensure that our students are probably the students that industry would choose over others. Our students should be the students who will perform better at competitive exams at the next level. Our students are the ones who will probably shine out among the graduates that will come out in 2024. So for that, we will have many activities that will be planned for you. And I would urge all students, and if there are parents along with who are now sitting with students and listening to this, ensure that your child or your students, ensure that you participate in every activity that you can, because this will add value. Adds value not to only to your marks, I mean, as on the B.Pharm certificate, but it will add value to your development as also a human being. And if you're aware, today the AICT, that is the All India Council for Technical Education, Pharmacy Council of India, in fact, the Ministry of Human Resource Development is focusing a lot on the personal development of the student community. And there is also a course which is there which students need to undergo. We will be sharing those links with you. It is on the human relations, universal human values, which as students probably you focus only on the technical aspect of your curriculum. You focus only on the, I would say, on the technical part, on the on your subjects, and you forget about how you need to collaborate with and how you need to work in a team, how you need probably the skills that you will require to work with or be with people in once you go to the industry, once you go maybe to another institute, probably you go to another country. So this is very essential for you to develop, and this is the time during the 4-year period while you will be studying pharmacy, while you'll be studying all the various subjects, you also need to build up and develop these skills, which will become very essential for you once you are out into the world and you get into the competitive world outside, where probably your B.Pharm certificate will get you the first placement, but progress from there on is going to be what you'll be learning throughout. So there is also a concept known as lifelong learning. So this lifelong learning is not just relating to the course that you study, not relating to the program, the curriculum that you study, but how you develop yourself as a human being. So knowledge, skills, values, all of these are very essential to bring up that holistic development in you as a student. And that is something which we at St. John will focus on, because yes, we have had toppers, we have had university rankers, and they're all doing well. And besides being the topper, you also need to know the skills that you will learn of how to get along with people, how to work in a team. All that will benefit you as you progress along the career ladder in your future career. So that's very important. And with that, I would once again welcome all of you. I hope to see you all see you all in the college campus as soon as possible. And we will be arranging sessions for you, arranging practicals. Details about when we will be able to begin with practicals will be communicated to you shortly. We are waiting for further instructions from the university. But follow the instructions of the faculty members, your class in charges. They spend a lot of time in preparing a lot of content, curriculum for you. So make the best use of the time. And as I mentioned, though you may find this first year a very tight schedule that is planned for you, but remember, we want you to complete it within the given span of time, so that you do not lose out. If other universities complete their work in time, you would probably be late for admissions when you go to your post-graduation. And we don't want that to happen. Hence, University of Mumbai has also decided that we will probably have a little condensed semester, and the topics that are to be taught, everything will be discussed with you. So comply with all of these.
You will be explained by all the rest of the faculty, all the other things that you need to know once you're in college. And surely once you are back here, once you come back to the college in regular mode, we will definitely meet you once again. We would also be calling a meeting of parents so that, we get to understand, because equally important is support that we require from parents to ensure that you, dear students, will be able to achieve what you are planning for in the next 4 years. So all the best, and do well. And we'd like to see a wonderful graduation result at the end of 4 years. Thank you.
क्या लग रहा है? हाँ वही है, हाँ प्रेजेंस का। और सर का वीडियो भी ऑन कर दो।
Good morning, everybody. Can you hear me? Please reply. Am I audible? Anybody can reply. Just say yes or no. Good morning, everybody. Can you hear me now? Please reply. Anybody can reply, just say yes or no, so I can start with the presentation. Can you hear me? Okay, so we'll start with the presentation. So first I welcome all the students to St. John Institute of Pharmacy and Research. I heartily congratulate you all for taking the admission to this institute and selecting for pharmacy profession. Now as you are in the first year, you will be coming across with different types of subjects. So basically we have 4 main subjects like pharmaceutics, pharmaceutical chemistry, pharmacology and pharmacognosy. And other subjects are the extension of these subjects, or they are the part of the main 4 subjects. So I am here to describe you the details about pharmaceutics subject. Pharmaceutics. Pharma means the knowledge of pharmacy or the knowledge of science, and sutics means formulation or preparation. So in short, we define pharmaceutics as the science of formulation or the science for preparing the different medicines or dosage form. So for preparation, what sort of knowledge is required? What sort of facilities is required, raw materials are required, and what precautions you should take for preparing a particular medicine, that all is included under the pharmaceutics. Now, as far as the first year syllabus is considered, you are going to study the first part of we cannot do like a simple lecture what we are taking in the class.
That is going on, but rest of the students are going to
Tell everybody to join the Google Meet, no? बरोबर 100% कमी होती. यूट्यूबचं भाडं काय तो काढला.
Sometimes click the button on the toolbar Google Chrome, Toolbar, you know?
आता डायरेक्ट देतो मी हेडफोनचा कौतुक पण करतात
So, can you go?
काय हेडफोन टेस्ट कर हेडफोन टेस्ट करना हेडफोन टेस्ट करना Okay, so coming back, there was some technical problem. So I am here to discuss about the details of pharmaceutics subject. I will just go back and repeat what we have discussed. We have seen that there are four main branches of pharmacy: pharmaceutics, pharmacology, pharmaceutical chemistry, and pharmacognosy. And now we have a new branch coming or emerging, that is the quality assurance for the evaluation of the drugs. Now let us see what is pharmaceutics. So it is the branch of pharmacy which is dealing with the preparation, formulation of a particular dosage form. Means how we can convert our raw materials into a final dosage form or final preparation. As a fresh student, you know different dosage form which you have used in our daily life. We all know what are tablets because everybody has taken the tablets at least once in your life. We know what are capsules because everybody has taken the capsule. We know what are injections, what are effervescent powders, what are suspensions, emulsions, ointments, paste, and we also know different cosmetics. So how these tablets are prepared, how an injection is prepared, how a suspension is prepared, that science or that technique of preparation is called as pharmaceutics. So in short, we are converting our raw materials into a final dosage form. So when I say dosage form, I am talking about a particular preparation like tablet, capsule, injection, ointment, paste. So that we are called as a dosage form in our pharmaceutical language. Now, what is important in pharmaceutics is the preparation knowledge, the art, the formulation techniques, the knowledge of packaging materials, and also the knowledge of evaluation. So pharmaceutics mainly requires the knowledge from all the branches like chemistry, pharmacology, pharmacognosy. We have to study the pre-formulation, we have to study the discovery of the new drug, and then we can convert that raw materials into the final preparation. So this is a very interesting topic, how to prepare a tablet, how to prepare an injection. Now as you are in the first year, you are going to deal with the basics of pharmaceutics. I am not going into the syllabus because you all know the syllabus of pharmaceutics, and that details will be also explained by the respective faculty in the afternoon session. I am telling you about the steps what we are going to take to cover the pharmaceutics. So first year you are going to study the basic dosage form. Basic means which are simple, which are used in our day-to-day life, like cough syrup, linked acids, solutions, some effervescent powders, or oral rehydration salts. You are also going to see some dispensing concepts, how to weigh the raw materials, how to do the mixing, how to perform the packaging. So all that basic knowledge you will get in your first year. You will also study the different aspects of formulation in second year, that is the pharmaceutical engineering subject. So which will give you information about different equipments, different machineries which are used for preparation of the dosage forms. Now as you move further to second year, you will study the microbiology, you will study the physical pharmaceutics, some physical properties of the raw materials of the dosage form. And when you go to the third year, you will come across with the main dosage forms like tablets, capsules, aerosols, suppositories, cosmetics, or some of the semi-solid preparations like paste, creams, ointments. And when you go to the final year, you will be coming across with the injectables, ophthalmic products, microencapsulation, or novel drug delivery system, or the manufacturing facilities, the evaluation aspects. So in a 4 years duration, you will be coming across with all the dosage form, including cosmetic science, including the engineering aspects, including microbiological and biotechnological aspects, and also the quality assurance of all the preparations. Now pharmaceutics also you can take as a specialization when you are going for the postgraduate, where you have to prepare a dosage form as a part of your research and you have to evaluate. So all your basic knowledge of undergraduate you can put into the postgraduate to prepare the dosage form of your choice falling under the novel category or a unique category. So on the slide you can see the definition of pharmaceutics. The technical definition is that pharmaceutics is the discipline of pharmacy that deals with the process of converting our raw materials into the conventional preparation. Conventional means old preparation, what we are using from years together, like tablets, capsules, injections, or into a novel drug delivery system which can be administered safely and which will be effective to the patients. So in short, it is called as the science of dosage form design. Now you can see here the different dosage forms. The diagrams are put on the slide, the diagram of capsule, the diagram of suspension, the diagram of the emulsions, what the bottles, we can see the primary packaging materials. This is the definition. Then you can see the diagrams of ophthalmic products, cough syrups or linctuses, Then you can see the diagrams of our inhalers, meter, dose or dry powder inhalers, tablets. Then we have the different semi-solid preparations and the injections. So these are our conventional, means regular preparations, what we use day to day as a part of medicine or as a part of cosmetics. Now this slide explains you about the different allied subjects which are related to pharmaceutics or formulation. So industrial pharmacy 1 and 2 you are going to learn in third year, where you will study the preparation of tablets, capsules, suspension, emulsion and semi-solid preparation. Pharmaceutical microbiology is also the allied subject of pharmaceutics, where your microbiological aspects will be covered under that, how we are converting or utilizing the pathogenic bacteria, non-pathogenic bacteria, and how we are preparing the vaccines and other preparations. Engineering deals with the industrial perspective, where you will study the mixing operation, drying, evaporation, packaging and other industrial operations which are required to prepare the dosage forms. Pharmaceutics 1, 2, 3, 4. 1 will deal with the basics, 2 is our physical pharmaceutics, 3 is our physical pharmaceutics 2, and 4 is the pharmaceutics which will deal with the parenteral and ophthalmic or novel drug delivery system. Apart from that, you will also come across with pharmaceutical biotechnology, where protein and peptide drugs, they are converted into the preparations like insulin and some of the vaccines. Then physical pharmaceutics deals with the physical and chemical properties of the drugs like rheology or surface tension or colloidal properties. Biopharmaceutics is also the part of pharmaceutics, where you will study the kinetic parameters of the drug. For example, if you take a tablet, what happens to that tablet in the body, like absorption, distribution, metabolism and excretion. Novel drug delivery system is a separate subject you will come across, where you will study all the novel preparation like extended release, controlled release, where you can increase the dissolution or the bioavailability of the products. Pharmaceutical jurisprudence, where you are going to study the laws which are governing the pharmacy profession. So this is a different law related subject, where you will come across with different acts or different laws like Drugs and Cosmetics Act, Pharmacy Act, Drugs and Magic Remedies Act. So this subject is totally the law subject, where or forensic pharmacy, which is very important to study, because while practicing the pharmacy profession, you should know the different laws we are coming under. Cosmetic science is also a separate subject, where you will study all different types of cosmetics. Skin cosmetics, hair cosmetics, colored cosmetics and other cosmetics which are used in our day-to-day life. And last is our quality assurance. So all the evaluation, regulatory aspects of the dosage form, how to import, export, what is the licensing procedure, all that will be deal in the quality assurance part. So pharmaceutics is also divided into different subjects, directly or indirectly, and all these subjects you are going to study from first year to final year. So this was the short introduction from my side related to the pharmaceutics subjects. So just a short revision, that pharmaceutics means the science which will deal with the formulation, preparation, or it is a simple technique to convert our raw materials into the final dosage form. So what is the dosage form? It is nothing but any preparation like tablet, capsule, injection, ointment, paste. We all know this. Up to 12th standard, everybody has used certain medicines. So we know what are these medicines. But how to prepare that medicine, that discipline is called as pharmaceutics. So in coming theory classes or in coming practicals, you will come across with different aspects of pharmaceutics first, and you will also prepare some of the dosage form, simple dosage form, and you will get a very clear idea about the basics of pharmaceutics in the first year itself. So with this I will conclude here. If there are any queries, you can ask the queries. The session is open for the discussion. Yeah, I am waiting for your queries. If there are any queries, you can ask the queries. Okay, so I presume that there are no queries. So thank you for your patientful listening. Have a great day ahead. हेडफोन
Good morning, everybody. After pharmaceutics department, I will take you through. I am Dr. Bharat Dhokchole. I will take you through introduction to pharmaceutical chemistry department. So, ideally, the subject of pharmaceutics is new to all of you because you have never studied the subject, but chemistry as such, as all of you have already studied the subject in your 11th standard, 12th standard, and even some part of your school. So this subject, at least initial part of the subject, is not new for you all. Normally, we see that people have fear for chemistry, right? because as you see on the screen, there are structures. You should know the different big structures of, and whatever structures you have studied in your 11th and 12th, comparatively you will have bigger structures or larger structures that you will be studying now in pharmacy. Okay, but if you take interest in the subject of chemistry, it will be easier and subject in which you can score the marks. Okay, so we'll go through the introduction to the subject, what are different aspects or what are different branches in the pharmaceutical chemistry. So as you all know, you all must have studied chemistry actually, but here it is called as pharmaceutical chemistry. So as you have seen in the introduction of pharmaceutics, that there is a study of different types of dosage form, but whatever chemicals or ingredients that are used for making dosage form, chemistry of those ingredients and API, that is studied in pharmaceutical chemistry. So it deals with different aspects. If you want to summarize it in a line, it is study of discovery of new drugs or chemicals and their development into useful medicine. So as such, pharmaceutical chemistry is divided into 3 or more branches. First and most important one is organic chemistry. Okay, so organic chemistry, I think almost all of you have studied in your 11th and 12th standards. Then you have biochemistry, that also you have studied already. The additional part or the new thing that you will be studying in pharmacy from chemistry is pharmaceutical chemistry and pharmaceutical analysis. So we'll look at those different branches of pharmaceutical chemistry. First one is organic chemistry. Now organic chemistry, at least initially whatever you study, the syllabus is nearly same that you have studied in your 11th and 12th science. Okay, so it deals with the study of chemistry of organic compounds. You study synthesis of various organic compounds, including heterocyclic compounds, their reactions and their applications. Also, there is one subject which deals with, which is the general chemistry or inorganic chemistry, which deals with the compounds which are from inorganic material. Then another important branch, or you can say a core branch of pharmaceutical chemistry, is medicinal chemistry. Now here actually you study the chemistry of medicinal compounds. Okay, then you should know their structures, their nomenclature of the various medicinal compound. So as you have seen in the introduction to pharmaceutics, that in the dosage form there is an API, okay, or a chemical entity which is responsible for pharmacological effect. So you should know the structure of that entity, you should know its nomenclature, so that all aspects are studied in medicinal chemistry. Also you should know how the drugs are discovered, what are the different discovery methods, what are the mechanism of action of the drugs, how the activity of drug may change if you change the structure, that is called SAR. Then you study therapeutic uses of different compounds, their adverse effects. So all these details of all the medicinal compounds or all the therapeutic classes of compounds is studied in medicinal chemistry. Then another branch of medicinal chemistry is pharmaceutical analysis. Now in pharmaceutical analysis, you study, as the name indicates, it is analysis of medicine or either dosage form and API. So it is a branch which deals with the methods for analysis of drugs, chemicals, that is API, and also formulation. It includes the study of different instrumental and non-instrumental methods of analysis. So non-instrumental methods are like titrimetry or gravimetry. So you all must have done the titrations in your 11th and 12th standards. Same types of titrations are also done for analysis or assay of drugs. Okay. Then there are also instruments used for the analysis of drug APIs or formulation. So different types of instrumental methods are available, like potentiometry, pH meter, UV visible spectrophotometer, HPLC. You will all study details of all these instruments, their principles, their instrumentation part, and their applications in the study of pharmaceuticals in this subject. Also, analytical chemistry deals with both qualitative and quantitative analysis. That means you determine also what is present, that is qualitative analysis, and in how much quantity it is present, that is called quantitative analysis. But here the special emphasis is with respect to application in the research and industry. So these are all the things that you will be studying in pharmaceutical analysis. So as we have discussed, it is an interdisciplinary science actually. There are multiple involvement of multiple branches, like there is a role of biochemistry, inorganic chemistry, organic chemistry. So all these branches, they converge and their knowledge is utilized to study the medicinal compounds in the pharmaceutical chemistry. Actually, traditionally, it is said that medicinal chemistry or pharmaceutical chemistry is an extension of organic chemistry. Okay, so in organic chemistry, you study about the structure and nomenclature of the compounds, but in pharmaceutical chemistry, you study the same thing about medicinal compounds. So it involves aspects like synthesis, isolation, purification, characterization of all the organic compounds that are medicinal compounds. So pharmaceutical chemistry is a very key link between the chemical structure and its biological activity or its use. So normally the pharmaceutical chemistry scientist, or if the person does PG in pharmaceutical chemistry and when he works in the industry, he's called a medicinal chemist, and he plays a very crucial role in a drug design and development. Okay, whatever new drugs are discovered, that part is done under the pharmaceutical chemistry division, and then it is transferred to pharmaceutics department for development into a dosage form. So if you look at the job prospects in pharmaceutical chemistry, if you, not after B.Pharm, but if you do a post-graduation or a PhD with respect to pharmaceutical chemistry division, then you have a chance to work in drug discovery department, then as a production chemist, or in the pharmaceutical bulk drug manufacturing, in pharmaceutical analytical R&D and quality control department. These are few job prospects after you pursue your PG or PhD in pharmaceutical chemistry. So now we'll go through the different, as you are going through the PCI syllabus, we'll just go through the different subjects that you will be studying in from your first year B.Pharm to your final year B.Pharm. So these are the subject. In semester 1, you will be studying pharmaceutical analysis, 1 theory and practical. So you will be studying different methods of titrimetric analysis in this subject. Okay. So most of the titrations that you will be studying in these subjects are already that you have completed in your 11th and 12th science. Here you will be doing the analysis with respect to drug compounds or medicinal compounds. There is another subject, as I told earlier, which deals with the inorganic compounds. There are some inorganic compounds, like for example, antacids, which are used as medicine. So study of those compounds is done in pharmaceutical inorganic chemistry, and also synthesis or analysis of these compounds. It is studied in pharmaceutical inorganic chemistry practical. Then as you move to the semester 2, there is pharmaceutical organic chemistry 1 and pharmaceutical organic chemistry practical. So as we have already seen, you study different methods of synthesis of organic compound, their reactions and their applications in this compound. And in practical, you will be studying how to identify the organic compound, what are different tests for identification of organic compound, and how to synthesize these organic compounds. Then next subject in semester 2 is biochemistry. In biochemistry, you study different aspects of carbohydrates, proteins, lipids, and how they are used as a drug targets or how they are relevant when you study the medicinal chemistry. Then in semester 3, you study organic chemistry 2. So here you will be studying heterocyclic compounds. Okay, their synthesis, their reactions, and their applications. Okay. Then in semester 4, you begin with the medicinal chemistry 1. So actually you start studying medicinal compounds, that is their structure, their nomenclature, their SAR, their mechanism in medicinal chemistry 1. And medicinal chemistry 1 practical involves the synthesis of medicinal compounds. Then in semester 5 you have medicinal chemistry 2. So you study another therapeutic classes of compounds in medicinal chemistry 2. In semester 6 you have medicinal chemistry 3 and medicinal chemistry 3 practical, where you will be studying the synthesis of drugs and drug intermediates. Then in your semester 7 you have instrumental methods of analysis and it's practical, where you will be studying all the instrumental methods of analysis, and practicals on which you will be doing analysis of these compounds. In semester 8 there are no core subjects, but you will be having 2 elective subjects, that is computer aided drug design and advanced instrumentation techniques. So these are all the subjects that you will be studying in your pharmaceutical chemistry department from your semester 1 to semester 8. See, normally there are students who are afraid or who are having fear of pharmaceutical chemistry, because there is a fear, or there is people feel that you have to study big structures and it is difficult. But if you study seriously from your semester 1 and if you are clear with all your basics in organic chemistry, it is easy for you all to study pharmaceutical analysis and pharmaceutical medicinal chemistry and whatever other subject you study in your higher semester. So I hope all of you will not have any fear of chemistry, and you will start loving and liking the subject, and that will what make the subject easy for your study. Okay, so that was all about introduction of pharmaceutical chemistry department. If anybody has any doubt, you can ask it now, or we'll proceed with the introduction to Department of Pharmacology by Dr. Deepak Bharti. Anybody has any question?
No.
Okay, if
No,
I
sir.
presume that
No, sir.
okay, so
Sir,
we'll
second
proceed
year
to
क्या
the
बोला था
introduction
आपने?
to pharmacology department.
Sir. Sir.
Yes.
सेकेंड ईयर में कौन सा सब्जेक्ट है?
Second year is pharmaceutical organic chemistry 2 in semester 3, and sorry. Yeah, you want subject in second year, right?
Yes, sir.
Yeah, I will just, yeah, these are, you can see there is pharmaceutical organic chemistry 2. Actually, your pharmaceutical organic chemistry 1 is already done in semester 2. So in semester 3, you have pharmaceutical organic chemistry 2, which involves study of organic compounds in semester 3. And in practical, you have same compounds, the identification of organic compounds. In your semester 4, you have pharmaceutical organic chemistry 3, in which you study heterocyclic compounds. And in semester 4, then you begin with medicinal chemistry, in which you study the therapeutic compounds, okay? Chemistry of medicinal compounds. Then in your semester 4, medicinal chemistry 1 practical, you begin with the synthesis of medicinal agents. Okay, so there is actually synthesis practicals. Is your doubt clear?
Yes, sir.
Okay. So now we'll proceed to Department of Pharmacology.
Good morning, students. Am I audible?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
I hope the screen is visible and my presentation is visible on your screen.
Yes, sir.
Students, I am taking you to the Department of Pharmacology. I am Dr. Deepak Bharti, head of the Department Pharmacology. If you see the cartoon in front of you, there is a pharmacologist staying at the door and saying, "Oh, no, this is not where you study to be a farmer." So you are taking a course where you are not going to be a farmer, but you are going to be a pharmacist. So, and you will be a part of healthcare system, and you are taking care of health of the peoples of the nation. So you have to be very careful while dealing with the drugs. And this pharmacology is the branch which will deal with the drug. And that is why it is very important to be conscious whenever you are studying the drugs, their desirable and undesirable effects, their side effects, uses of the drugs, and that is why we have to study different subjects into this department, that is pharmacology. So we'll just, if you break the word pharmacology, you will get pharmacon and the logos. The pharmacon is nothing but the drug, and this whole branch is dealing with the drug only, as we have already gone through the pharmaceutics and pharmaceutical chemistry, where in pharmaceutics you will deal with the different pharmaceutical formulations. You will deal with different pharmaceutical formulations like tablets and capsules and parenteral formulations. In pharmaceutical chemistry, you are going to synthesize new chemicals, new drug moieties in the pharmaceutical chemistry labs, and then you will go for their analysis and all that study. But in pharmacology, you are going to deal with the study of effect of that drugs, right? So it's a science of the drug which deals with the study of desirable and undesirable effects of the drug. Now, desirable means the drug which is taken for what, right? If you are taken the drug for hypertension, then that is a desirable effect. But along with the desirable effect, drug is also showing some undesirable effects. So, study of all the intended and unintended effects of the drug will be there in the pharmacology. So, it's a crucial branch into the pharmacy, and you will go through different drugs. Whichever drugs are available for the treatment of various diseases currently, all will be studied into the pharmacology. So pharmacology is the study of drug and their actions onto the body. So there are different types of systems into the body. There are different organs involved into the body. So what are the different effects of your drugs on different organs and different systems and their physiology, that will be studied into the pharmacology. So pharmacology is the study of drugs and their actions onto the body. There are different branches of pharmacology also. If you see on your slide, pharmacology is divided into 5 branches: pharmacokinetics, pharmacodynamics, pharmacotherapeutics, toxicology, and then it will come to the pharmacy. So whenever you are taking the drug into the body, if you are administering the drug into the body, your body is doing something with the drug, right? There will be absorption and distribution and metabolism and excretion of your drug, because anything which you are taking inside the body has to get eliminated out of the body. So your drugs will also, if they are administered, they will get eliminated, and so there will be, your body is also doing something with the drug. That is, the study of that is called as pharmacokinetics. So pharmacokinetics is a branch of pharmacology where you have to study what your body is doing to the drug. There are different functions your body is doing, like absorption and distribution and metabolism and excretion of each drug. We have to go through in this branch, pharmacokinetics. Second branch is pharmacodynamics. Now your drug is taken. Now drug is also doing something to the body, which will show the biological responses, biological effect, pharmacological effects. That is, the study of that is called as pharmacodynamics. What drug does to the body is pharmacodynamics. So these are the 2 important branches of pharmacology: pharmacokinetics, what body does to the drug, and pharmacodynamics, what drug does to the body. After that, we have to study the uses of the drug, pharmacotherapeutics. In which condition, which drug to be taken. That is also important, because there are different drugs used in the different conditions, in different pathological conditions. One drug can be used in different pathological conditions. Though you have to make a choice of drug while prescribing the drug, while administering the drug to the patient. That study of that type of study is called as pharmacotherapeutics. The study of uses of the drug into the patient for particular pathological condition. The indication is there for a particular class of the drug, which drug to be prescribed, to be studied into the pharmacotherapeutics. And along with that, we are not studying only the pharmacological effects of the drug, but it is equally important to study the toxic effects of the drug, side effects of the drug, adverse effects of the drug. That all the side effects, toxic effects, adverse effects of the drug are studied into the toxicology. It's a very important branch of the, nowadays you are going through the COVID-19 situation, and everyone is aware of there are some vaccines are introduced, and many news channels are reporting that these vaccines have this side effects, that vaccine has that side effect. So that study of this type of adverse reactions by the new drugs, study of the adverse effect of the any old drug, that will be studied into the branch of toxicology. That is also a very important branch of pharmacology. So with toxicology is nothing but a study of adverse effects of the drug, study of toxic effects of the drug. That drug may be a new drug, that drug may be an old drug, but the monitoring of your adverse effect is very crucial stage, and that is done into the toxicology. And finally, drug should be administered, drug should be dispensed into the suitable dosage formulation. This pharmacy is actually a department of pharmaceutics branch where the formulation of different formulations are prepared and dispensed to the patients. So this is also a very important branch of pharmacology. So if you connect, there are four important subjects into the pharmacy. One is pharmaceutics, pharmaceutical chemistry, pharmacology, and another one is pharmacognosy. So each subject is interconnected with each other, where the new drugs were discovered into the pharmacology. That source of the drug may be from the pharmaceutical chemistry lab synthesized, or it may be isolated from the natural sources like the plants or the other minerals, and that will be studied onto the animals and humans by the pharmacologist. That new drug isolated from the plant sources or synthesized into the pharmaceutical chemistry will be evaluated onto the animals for their toxic effects and the beneficial effects. Then it will be studied onto the. Someone's mic is on. Please mute your mic. That will not disturb others. Should we continue? So this pharmacologist will evaluate the effect of drug on animals and then onto the humans. Pharmacologist will study the effect of drug and side effects of the drug on human volunteers, and then drug will be forwarded to the department of pharmaceutics, where it will be formulated in different formulations, and then it will be dispensed to the patients for the administration. So we'll just go through the different subjects which are there for your 4-year course in semester 1 to semester 8. In semester 1, we have Human Anatomy and Physiology 1, which is a theory portion. Human Anatomy and Physiology practicals is also there for 4 hours. Now see, if you want to see the effect of the drug, desired and undesired effects of the drug onto the body, then you have to study the different parts of the body, different systems and their physiology. And for that, first 2 semesters, in first year, first semester 1 and semester 2, you have to study Human Anatomy and Physiology in detail. Along with that, some practicals also you have to perform on Human Anatomy and Physiology. In semester 2, that semester 1 Human Anatomy Physiology 1 is extended to the semester 2. Remaining systems will be studied in semester 2 with the Human Anatomy and Physiology, and practicals of Human Anatomy Physiology also in semester 2. Along with studying the drugs, different anatomy part and physiology of the body parts, you have to study different diseases. So which type of diseases, which systems are affected by the particular diseases, etiological factors of the disease, and how pathophysiology of that particular system is affected due to the disease conditions, that will be studied in pathophysiology. Okay, so we'll just go through this Human Anatomy Physiology. This subject is designed to impart a fundamental knowledge on the structure and functions of various systems of the human body. It also helps in understanding both homeostatic mechanisms as well as functioning physiology of the body. The subject provides basic knowledge which is required to understand various disciplines of the pharmacy. It will not only required for studying the pharmacology, but it will also require to study how formulations are also affecting the particular body system and body organs. So all the disciplines require basic knowledge of Human Anatomy Physiology. Objective of this course is explaining the gross morphology, structure and functions of various organs of the human body. This course will also describe the various homeostatic mechanisms and their balances and imbalances. In practicals, you can perform different hematological tests like blood cell count, like WBC count, RBC count, hemoglobin estimation, bleeding time, clotting time, as well as you also allowed to record blood pressure, heart rate, pulse and respiratory volume also. During the practicals, you can perform this type of practicals. It will also appreciate the coordinated working pattern of different organs of each system, how cardiac system is coordinated with the respiratory system, how central nervous system coordinating your heart, cardiac system and renal system. So this coordinated working pattern will also be studied into the human anatomy physiology 1 and 2 as well as in practical 1 and 2. Second, semester 2 having a pathophysiology, and this course is designed to impart a thorough knowledge of relevant aspects of pathology of various conditions with reference to its pharmacological applications and understanding of basic pathophysiological mechanism, various disease condition, the etiological and pathogenesis of some selected state diseases. They are also how that pathophysiological condition affects the normal physiological functioning of the system. Everything will be covered in the pathophysiology. Then only this basic knowledge of human anatomy, physiology 1, 2 and pathophysiology is very essential to understand the different drug and their mechanism in pharmacology when you will start with the pharmacology 1, 2 and 3 in the semester, next semester. That is semester 4 we have pharmacology 1 and pharmacology 1 practical. Semester 2 we have pharmacology 2 and pharmacology 2 practicals, and in semester 6 we have pharmacology semester 6 we have pharmacology 3 and pharmacology 3 practical. So semester 4, 5, 6 you have pharmacology 1, 2, 3 and 1, 2, 3 practicals, theory part and practical parts. During this pharmacology 1, 2, 3 theory and practical work, you have to go through all the drugs which are available currently in your healthcare system, all the drugs which are affecting all the systems, all the disease conditions. And in semester 8, we have 2 elective subjects given by Pharmacy Council of India's syllabus. One is pharmacovigilance and another one is experimental pharmacology. Now what we will going to study in pharmacology is in theory and practical. The main purpose of subject is to understand what drug does to the body, like living organism, and how their effects can apply to the pharmacotherapeutics. So how your in summary we can say we have to study pharmacokinetics and pharmacodynamics of all the drugs during the pharmacology 1, 2 and 3. The subject will cover information about the drugs, their mechanism of action, physiological effects, their biochemical effects, as well as absorption, distribution, metabolism and excretion of the drug, along with adverse effects, clinical uses of the drugs, drug interaction, dose, contraindications and routes of administration of different drugs, drugs acting on different systems. This we have to study through this pharmacology 1, 2 and 3. Once you are completing the course, you should be able to explain the mechanism of action of the drug at organs, at tissue levels or macromolecular level. You should apply basic pharmacological knowledge in prevention and treatment of various disease conditions. Once you are thorough with the pharmacology, all the courses, and observe the effect of drugs on animals by the simulated experiment. This we will cover in pharmacological practicals. This is the practical work, like we have to study the effect of drugs onto the animals. Since animals are not allowed into the educational institutions, so we will study the effect of drugs on the simulated experiments and demonstrations. And also we have to appreciate the correlation of pharmacology with other biomedical sciences, like pharmacology is applicable to other biomedical sciences, like toxicology is also there, biology is also there, different tissues and biological tissues, the effect of drug we have to study on that also. Next one is elective subjects which are there into the semester 8. 2 elective subjects are there. One is pharmacovigilance and second one is experimental pharmacology. In pharmacovigilance, which is, I hope you know that adverse drug reaction, monitoring, detection, monitoring and reporting of adverse drug reaction is continuous process. Once drug is launched, drug is marketed into the general population. And in that case, the continuous process is reporting of adverse drug reaction is happening into the, and that branch is called as pharmacovigilance. So this pharmacovigilance subject will give a opportunity for the students to learn about the development of pharmacovigilance, basic terminologies which are involved into the pharmacovigilance, global scenario, how the global what is the global scenario of pharmacovigilance in India as well as across the world. This subject will also train the students on establishing pharmacovigilance program into the organization, into the institution where you are going to work. Various methods that are used to assess the adverse drug reaction, to report the adverse drug reaction, generate the safety data and signal detections, and also regulatory aspects of the pharmacovigilance. This is not only required reporting and monitoring, but there are some regulatory aspects also. There are different rules and regulations by the governing authorities that we have to follow while doing this pharmacovigilance. Everything will be covered into the pharmacovigilance, which is an elective subject in semester 8. Second elective subject for semester 8 is experimental pharmacology. Now, this subject is designed to impart a basic knowledge of preclinical studies. I hope you know this preclinical study means where the animals are used to study the effect of drug. When animals are used to study the effect of the drug is called as preclinical studies. Few terminologies may be new to you because as it's a first year only, but as and when you will be in the next semester, these terminologies will be a regular terms for you. So these experimental animals are used. So this subject is introduced in the course because you should, whenever you are passing out with your undergraduate course, getting a degree of B pharmacy, you should have a basic knowledge about the preclinical studies and which experimental animals, how to design the research, how to conduct and interpret the data collected from the animal studies while doing the experiments onto the animals. That will be there into the experimental pharmacology. This will be once you are completing the course of experimental pharmacology, you will be able to use your knowledge in the on commonly used laboratory animals. Also, able to demonstrate the various screening methods used for preclinical studies. There are different methods used to screen the drug onto the animals that are called as screening methods. So once you are done with this course, that will give you an ability to demonstrate these methods also, and you will be able to demonstrate the importance of biostatistics and research methodology because you have to interpret the data obtained from the studies on the animals, and then you can apply it onto the humans. For that, some biostatistic methods and some research methodology concepts have to be applied onto the data obtained. So this will be also there into the experimental pharmacology, and you are able to design and execute the research hypothesis independently without anybody's help once you will complete this course. So these are the subjects of pharmacology from semester 1 to semester 8. You only don't have any subject in semester 3 and semester 7. Otherwise, in each semester, there will be a subject from the Department of Pharmacology. So if you have any doubts, any clarifications you require, you can ask me now. I hope I was connected with you. Yeah. In any clarification, any doubt you want? Otherwise, we stop the presentation and then we will move to the next Department of Pharmacognosy by Mr. Abhijit Puri. Hello.
No, sir.
Okay, thank you very much. I request Mr. Abhijit Puri to start with Department of Pharmacology. Thank you.
नहीं हाँ
कुठली आहे तुमची
प्रेझेंटेशन
Yeah, good morning everyone. Am I audible to you?
Yes, sir.
Yes, sir.
Okay. So myself, Mr. Abhijit Puri, Assistant Professor in Department of Pharmacognosy. So I'll be taking you through the introduction to pharmacognosy subjects and different subjects under this pharmacognosy. So what is pharmacognosy? Pharmacognosy is actually a study of natural products or natural drugs from different origins. Okay, that is from mineral origin, from plant origin, from animal origin, their physical properties, their chemical properties, their biological properties, then their methods of harvesting, cultivation, collections, then its botanical aspects. Then I'll be taking you through some of the in this subject. I'll be explaining you about some of the extraction isolation methods which is there. Then about the different under the subject different quality control parameters, and all this will be looking into this subject now. So here, as I told you, pharmacognosy, it's a subject which is dealing with the study of physical, chemical, biological or biochemical and its biological properties of natural products or natural crude drugs which is obtained from different sources. The sources as in form of plant, animal, mineral, marine, microorganisms, some hormones, some tissue culture, plant tissue culture derived products. So this subject pharmacognosy is actually dealing with this different aspects, that is taxonomy and botany, which is the base of this subject. As you know, in this pandemic now, you could have seen various formulations which were available in the market, or everybody was taking some herbal ingredients. We're going to the some herbal ingredients. For example, giloy. You could have seen this giloy in various formulations in the market, different ads. Avla, okay, all this type of then muleti, then ashwagandha. All these are immunomodulator drugs which were being sold on a very larger scale, or I mean to say they were formulated and then they are available because they are thought to protect you against several disorders, diseases or different microorganisms, or they are used as an antiviral formulations. So pharmacognosy is it's taking up like I can say it's sharing most of the part of the formulations. So nowadays you can find there are many new formulations which are coming up by using different pharmaceutical techniques, nanoproducts, nanoparticles, using nanoproducts, or there are different technologies which have been involved, and then where they have incorporated herbal ingredients and that formulations have been reintroduced into the markets. So such all formulations, before formulating, before this Dr. Bharti, he has explained you how a herbal drug is used for checking its activity against different animal models, or how pharmacological or he has told you about pharmacological aspects of different herbal drugs. Dr. Asne has explained you about pharmaceutics, where different ingredients, herbal ingredients are used, basic ingredients which are used to prepare formulations. They are obtained from the natural sources. So that natural sources we are going to study it here. Then Dr. Bharat has told you the aspects of some medicinal chemistry, in which the major part, like I mean to say, the semi-synthetic product which is formed from natural sources, or a lead molecule which is there, it's obtained from different natural sources, and then that lead molecule is modified into a semi-synthetic product and it comes into a market in form of a drug. So this pharmacognosy is actually based on traditional systems of medicine, different Ayurveda, Siddha, Unani, then homeopathy. After all this like all these traditional systems are being used in India, or it has been largely accepted throughout the world. WHO has recommended these practices for various disorders, diseases, and now by using their basic principles, we have a subject where all that principles have been included in the subjects, and now you're going to study that subject in more detail. So what all you need to study in this subject is about a herbal drugs, about a non-herbal, that is in form of animal product or mineral origin product. You need to study its origin, that is the origin will be studied through its taxonomy, from where is its taxonomical source, under what class, under what phyla, under what category it falls. Then its botanical aspects, whether it is organized drug, whether it is unorganized drugs, that is whether it has a specific structural because this what I mean to say here is whether it has some specific structure to it, or whether it is an unorganized form of drug, where both unorganized and organized drugs, they are largely used for several formulations. Then distribution of this organized drugs, unorganized drugs, like your organized drugs can be different examples, what I told you just now about muleti or giloy or avla. These are organized drugs, and unorganized drugs are like different honey, this kind of volatile oils or edible oils. All this comes under unorganized drugs. So by using them and preparing an formulation from them, for that first you need to know from which plant it is being extracted out, from which plant it is being isolated. For that purpose you need to study the plant is from which origin. So throughout the world, different plants in different regions they grow. So it's study of that geographical sources, study of the regions from where that plant is obtained, because particular plant grows well in that particular environment or in that particular region, so that you can use that plant because it synthesizes that constituent only in that regions. So distribution of natural products throughout the world you need to study. Then you need to study different methods of cultivation, little bit farming. Your farming like techniques you have to apply here, or I mean to say you will be studying little bit farming of cultivation, collection, harvesting, processing of crude drug, storage of crude drug, and in that then there are different methods, soil conditions, environmental conditions, what type of fertilizers you have to apply, what type of irrigations you have to supply for the plant, all that, because they have to synthesize this phytoconstituents in them. So you will be studying description of plants under various categories from each phytochemical categories, then the active constituents from different sources, as I told you just now before this. So that active constituent of what category actually it is, that you are going to study it here. Then how they are biosynthesized in the plant, how they are formed into the plant, where they are stored, in which cells they are stored into the plant. Okay, that biosynthesis, like starch, it is biosynthesized. It's a polysaccharide which is biosynthesized and stored in several cells, like amyloplast cells, they are stored. So like that you are going to study the biosynthesis of different constituents, and then their storage in different plant part. Then different plant parts which is being largely used, so that, like for example, leaf, mint, digitalis, senna, licorice, root in form of licorice, ashwagandha root, shatavari root, seeds, different examples of seeds, coffee seeds, castor seeds, then different examples of bark you will be studying. Like there are many examples which you will go through. There are more than 200 drugs which you have to study it in your entire syllabus. It's cultivation, collection, I told you, it's storage, different processing during storage, then the containers in which you have to store it, at what temperature you have to store it, all that properties you have to study it here. Physical properties of the drug, chemical properties, its biological properties, that is its biological action on the different animals, or pharmacological actions of the crude drug you are going to study. Then you will be also studying its different therapeutic uses of the different natural products. You will be also studying to isolate that compound, because one plant will contain more than, not less than, I mean to say many number of phytochemicals. So from that, only one particular ingredient, removal of one particular ingredient or separation of one particular ingredient, and then studying its biological properties. So this all you will be studying under pharmacognosy. So here the subjects which is there under pharmacognosy is in second year B.Pharm, in semester 4 you have pharmacognosy and phytochemistry 1 theory and its practical. And they are interrelated. That I'll tell you how they are interrelated. Then in second year, in semester 5, you'll again have pharmacognosy and phytochemistry 2 theory and its practical. And then in third year B.Pharm, you have herbal drug technology. That is also, you'll have a theory and practical for herbal drug technology. And it is like from the basics it started. In SY B.Pharm, that is in pharmacognosy 1, what you are going to study is the subjects. It will involve all fundamentals of pharmacognosy. That is its scope, different scopes of pharmacognosy, where pharmacognosy is applied in which fields. It's applied in biotechnology, it's applied for microbiology, it's applied for formulations. So in each different field it is applied. So that applications we'll see. Then different class of crude drugs, from where they are obtained, that classification you will be studying it in this subject. Then you will also study a very important parameter as to how to identify a crude drug, or whether it is medicinal plant, whether it is toxic plant, whether it is poisonous plant, or whether it is of an ornamental type, where it is of non-use. So all that identification. Then you will study evaluation, how to identify the adulterants, that is in form of some malpractices is done, or some kind of unwanted substances are added in the crude drug. You will be able to evaluate that. You will be evaluated to different types of, you will be known about, or you'll be taught about different phytochemical aspects of the crude drug, then its medicinal properties, therapeutic uses. So under this first pharmacognosy 1 or pharmacognosy and phytochemistry 1, you will have to study this different aspects, and you should on completion of the course you should be able to know the techniques for cultivation, collection, that is basic farming. I mean to say that different for the different crude drugs from different regions, from different parts of the world, not only India but different parts of the world. That also you are going to study it here. Then you will be able to know the crude drug, their uses, their chemical natures, class, phytochemical class. You'll be able to evaluate the crude drug. Like you'll be, after studying this, you'll be familiar with evaluations of different aspects of evaluations of crude drug. Then you will be able to carry out evaluation based on its botanical characters, that is microscopical character, its external features, morphological characters of the crude drug. So this will be a basic pharmacognosy which you will be studying in pharmacognosy 1. And under this, then when you have a practicals related to this, the practicals are also same, some botanical aspects of your different crude drugs. So you will come to know morphology of crude drug, you will come to know different types of crude drug, that is you will be studying it in practicals. Then you will be evaluating the crude drug. Okay, all that evaluation parameters you are going to study, ash value, extractive value, these are different parameters, okay, under which you are going to study it in the practicals. So it will be interrelated. What topics you are going to study under this pharmacognosy and phytochemistry 1, same you will be having it in your practicals to perform the practicals. Then pharmacognosy 2. Now in pharmacognosy 2, you will be now brought little bit closer to the actual, how the constituents are there in the plant, or how they are synthesized into the plant, how the chemical reactions, or I mean to say biological reactions, is occurring to form these constituents in the plant. So basics about some phytochemistry you will be able to study it here. You should have some basic knowledge about it, which you will be taking up from the other chemistry subjects, and then you will be like biochemistry and all the other subjects, you will be applying it here. So here secondary metabolite synthesis you will be learning how to now actually isolate the plant constituent, phytoconstituent, from a particular source of crude drug, not only plant but marine source or animal source, whatever it is. Okay, then to identify and use them for the further formulations, how to use, after isolation, after evaluation, now how to formulate it. So its formulation aspects you will be studying it here in this pharmacognosy and phytochemistry 2. Then its regulatory aspects also you will be studying related to formulations, because several different types of drugs which are there. For example, some drugs which comes under the Drugs and Cosmetic Act, some comes under Narcotics Act. So all that different regulations about the drugs you will be studying here in this pharmacognosy and phytochemistry too. And then it is also involving formation of phytochemicals or increase the yield of phytochemicals by using several tissue culture techniques. But that means you are going to actually study about how the constituents are synthesized in plant, and that is done it in laboratory, and then that is in biotech lab, you can say. And then that biotech knowledge or that basic tissue culture knowledge you can apply for further cultivation collection, or you can use that basic, like for example you can use that knowledge of synthesis of some kind of hormones in the plant by using different techniques of plant tissue culture, how to form transgenic plants, all that you will be studying it here. Then you will be studying interaction of drugs, herbal and drug interaction, food and herb drug interaction, herb and other ingredient interaction. So all this drug herbal interactions, all the interactions, because it is an phytoconstituent, it will interact with your normal allopathic drug or semi-synthetic drug or synthetic drug. So that the interactions in the body, because on simultaneously when we will be on a dual therapy, that is on some herbal therapy, I mean to say Ayurvedic or any other traditional systems of medicine, if you are taking along with that synthetic drugs, then it's interactions. So what you have to avoid, which type of drugs you have to avoid, which type of food you have to avoid, that all you'll be studying it under this topic. And then you will also study basic principles of traditional systems of medicine, each traditional systems of medicine. From that you will acquire a knowledge about use of different plant products by their from their formulations, how they were formulated in their traditional systems of medicine. Then upon completion of this course, what you will be able to do, you will be able to actually extract, you'll be able to know about extraction techniques, characterization technique of that constituents. You'll be able to identify the herbal drugs based on their phytochemicals. You will be able to classify them based on their phytochemical, based on the class of phytochemicals. Then you will be able to understand herbal formulations, how to prepare herbal formulation, what all ingredients are used in that herbal formulations, what are different processes are there for this herbal ingredient processing and then formulations. And or you will also be able to know about its evaluation of herbal formulations over there. Then herbal drug interactions, as I told you, will be able to understand which drugs to be taken with which herbs. Then you will be able to isolate, actually you will be able to know about separation of constituents and how to identify that constituents. And in the next year, that is in the third year, you will come more closer to the actual aspects of herbal drug technology, that is industrial method of formulations. Herbal drug technology, which will deal with different quality control of raw materials, its evaluation of raw materials, finding adulterants in the raw materials, guidelines for quality control of herbal drug materials, quality assurance parameters of herbal drugs, or herbal cosmetics, natural products, natural sweeteners, nutraceuticals, herb food which we take. They are called some food ingredients which are used as an for the purpose of health supplements, as in multivitamins or proteins. They are called as a nutraceutical type of compound. So you will study that nutraceutical formulations over here. Then you will come across some good manufacturing practices of how to formulate the crude drug by using good manufacturing practices, industrial aspects, its patenting, regulatory aspects, more detailed regulatory aspects according to WHO, according to ICH guidelines, how to evaluate the crude drug, what are its regulatory aspects, that you will come to know here. So you will understand about raw materials upon completion of course. It's different guidelines you will be able to understand of evaluations. You will use this knowledge for formation of some cosmetics. You will be able to apply this knowledge to form some nutraceutical compounds, or you will be able to acquire the knowledge about how to patent a herbal formulation or a herbal drug, or its good manufacturing practices you will be able to learn in this herbal drug technology. So these all topics or these all subjects you have in pharmacognosy. Okay, so anybody has any doubt or anybody wants to ask any questions? So the session is open for questions. No? Thank you all.
उसमें हो रहा है academic guidelines. Academic guidelines.
Share also.
Yeah. All right, students. So next session is by Dr. Govind Asne. He will be running you through the academics regarding the lectures, practicals, what not to do and what to do. So I request sir to start with the session. So he'll start presenting the slide. The presentation is visible to everyone.
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Yes, ma'am.
Good afternoon, everybody. Can you hear me? Please reply.
Yes, sir. Yes,
Yes,
sir.
sir.
Yes,
Yes.
sir.
Yes.
So I am again back to elaborate you on academic guidelines. So being holding the responsibility of academics, I want to make you aware about certain practices, certain rules and regulations which we are following in the institute. Now, as these guidelines are applicable for offline mode, means when students are coming to the college, these guidelines are prepared by thinking of the offline mode. But currently, as you are in online mode of education, some of the guidelines will not be applicable for online mode of education. So I will start with the presentation. Everybody can see the presentation. Please reply.
Yes, sir.
Yes,
Yes,
sir.
sir.
Yeah,
Yes, sir. Yes, sir. Yes, sir.
thank you. Thank you. Now, first, I will introduce you to all the departments, introduction to the faculty members, and most of the faculty members you may be knowing, but you may not have seen them because of this online mode of teaching. So let us start with the Department of Pharmaceutical Chemistry and Analysis. So we have our principal, Madam Dr. Savita Tauro, as a principal and a professor in pharmaceutical chemistry. Dr. Bharat, who is associate professor and HOD, he has given you the explanation on pharmaceutical chemistry department. Then we have Mr. Prashant Chaturvedi, who is also our training and placement officer. After me, the session will be followed by him. Mr. Avinash, Mrs. Dipali Nahar, Mrs. Sahaya, Mr. Ramesh Bhusal, Mrs. Ekta Thakur, Dr. Navnath Hathwate, Mr. Tanvir Singh Rajput, Mrs. Varsha, and Mrs. Saswati Panigrahi. So these are the faculty of pharmaceutical chemistry department. So some of the faculties, they are teaching to the first year. Some of the faculty you will come across in your second, third, and final year. And so you will have a good interaction with all the faculty when you will be coming to the college. Now going further to pharmaceutics department, Dr. Govind Astin, that is myself, Dr. Praveen Pawar, who is also professor in pharmaceutics, Dr. Norma, she is associate professor looking after mentoring aspects, Mrs. Rushali Gokhale, Mrs. Meeta Jain, Mr. Amol Golab, Mr. Sudarshan Kakad, who is taking the analysis, Ms. Dela, Mr. Prashant Dalvi taking your pharmaceutics, Mr. Melroy, Mrs. Nisha Kalail taking your pharmaceutics subject and also the coordinator of today's function, Mr. Prashant Shinde and Ms. Swapnali Shinde. So these are the faculty in pharmaceutics department. Some are taking for your first year, some you will have interaction in second, third and final year. Then third department what we have is Department of Pharmacognosy. So just Mr. Abhijit Puri has finished his pharmacognosy presentation and along with him we have Mr. Prakash Khandagle. Then Department of Pharmacology, Dr. Deepak Bharti, who has given presentation on pharmacology subject. Then we have Mr. Naneshwar, Mr. Jagan Nadar and Dr. Nitin Kumar Sen, who are taking your human anatomy and physiology subjects. Now this was the introduction to different departments what we have in the institute. We have total 31 faculty and some of the faculty will be your mentors in the coming days, or they will be mentors till you pass out or till they are in the college. Now going further, this is our main part of today's discussion, that is the academic monitoring and guidelines, how to monitor the academic activities. Now what comes under the academic activities? First one is to prepare the workload of faculty and also to prepare the timetable by the respective class in charges. So you know that you have Nisha ma'am and Varsha madam as your class in charges. So these two faculties will be looking after your timetable, looking after your academic schedule is happening day to day, whether all your lectures are happening, practicals are taking place. So any doubt, any difficulty regarding the theory and practicals will be handled by the respective class in charges. So the CR will be appointed, that is the class representative, and that CR will have a coordination with the class in charges, and then they will see that all the lectures and practicals are happening and the activities are conducted smoothly. Second function or the second activity is to supervise the regular academic activities as per the timetable. So apart from the regular timetable, we are conducting a lot of curricular and extracurricular activity, which are also the part of our academic calendar as well as the academic schedule. So activities may be related to the sports, cultural, It may be related to your industrial visit, or it may be related to some of the technical events which we conducted in and off the campus. So you can participate in the events which are conducted by the college, or also you can participate in the events which are conducted by other colleges, that we called as external participation. So we coordinate all these curricular and other activities, and we see that the students' participation is maximum in all these activity. Then we have the another role to play in the academics is to monitor the daily attendance of the students, the MIS entries, and the academic diaries. So this is applicable to the faculty. So we all know that as this is the online mode of education happening currently, the attendance is monitored of the students and it is directly captured in the MIS. But when the physical mode of teaching will start, we have a daily attendance sheet which is kept in the class. The physical attendance will be taken. That attendance will be entered into the attendance sheet. The topic will be entered by the faculty, and based on that entry, the attendance will be uploaded in the MIS. So all the students can monitor their attendance by logging into the MIS. So everybody will get a MIS ID and password. You, by sitting at home, you can monitor your attendance. Your parents can monitor the attendance. And so if there is any rectification, you can bring this rectification to the notice of the faculty or to the class in charge. Then we have periodic assessment of syllabus completion. So you all know that you have two periodic examination as per the PCI syllabus. So in one semester, you will be appearing for two periodic theory and practical examination and for one semester end examination. So how much syllabus to keep for first periodic exam? What will be the syllabus for second periodic exam? That will be decided and that will be communicated to all the students. Also, we take the feedback review of the syllabus completion. So whether the syllabus completion is happening in a right direction, whether the faculties are going too fast, too slow, whether all the subjects are covered within the stipulated time. All that things we monitored very frequently, and the feedback is taken from the class representatives or from the students directly or indirectly. Then we also monitor the lecture adjustment. So whenever a faculty is not available, that lecture will be adjusted by some other faculty, and we see that all the slots of lectures and practicals are conducted so that not a single lecture goes off or that slot goes vacant. Now this is possible when the offline mode is available. But online, if faculty is not coming on the 11th hour, then we cannot adjust the lecture because that faculty has to create the lecture in the calendar, link has to be created, it has to be communicated, it will take time. So 11th hour adjustment in online mode is little bit difficult, but prior adjustment will be done and we will see that all the lectures and practicals are happening smoothly. And last is our to maintain all the records, documentation which are related to the academic activity. Now this is what we called as academic monitoring. From the first day, that is from 1st February, when your teaching has started, we are monitoring the lectures, practicals, We are monitoring your attendance, syllabus completion, and we are planning that whatever the number of days are available, the syllabus will get completed in that number of days. Now in morning, Madam has elaborated on the academic calendar. Already you are late by 6 months, and so in a period of 8 months, you have to finish 2 semesters. So first semester will end by 30th April, and from 3rd May, your second semester will start. So all the two periodic exams, one semester exam of theory and practical will happen before 30th April, and from 3rd May, you will be coming into the second semester. So whatever the teaching learning days are available, 50 or 55, we have to utilize all these days to complete the entire syllabus. So depending upon the importance of the topics, so what Madam has also elaborated that important topics will be taught first priority basis, and then it will go further to complete the syllabus. Now, after discussing this academic monitoring, the next point is the attendance of the students. Now, attendance is not a problem when online education is going on because you can log in from the home. But this attendance will be a problem when offline education will start, means when you have to come physically into the college. So due to number of reasons, it will not be possible for the students to come every day to the college. I'm not talking about this COVID-19 situation. I'm talking about when the situation will become normal, or in the earlier part when it was a normal situation, it was compulsory for all the students to come to the college. So due to different reasons, if the attendance of the student is very less, we have a provision of sending the letters to the parents, and will also inform the students. So they will take appropriate measures and that attendance can be improved. So apart from you, we will be informing your parents also regarding your attendance, regarding your progress in the exams, regarding your all the other activities participation or overall growth from first year to final year. Your parents will be also informed about all the activities. So theory attendance and practical attendance, we are monitoring it separately, and whenever the attendance drops, goes below the prescribed as per the university rule, then a letter will be sent to you, and if the defaulters are there, then as per the university regulations, the further action will be taken as per the university provisions. So just see that your attendance will never drop. If you have any problem in the sense if you are sick, not feeling well, or if you want to go to your hometown, attend some functions, or whatever is the reason, you should always inform your class teacher. You should take the permission in writing. If you are sick, submit the medical certificate, or if you are attending the weddings, give the wedding invitation or whatever the document you have, or if you are participating any sports external, then that certificate has to be given, and so we will consider the attendance in these cases. But without any valid reason, if you are remaining absent, then the further what we have discussed, the letters and other necessary provisions that will be taken by the institute. Then we have the provision of conducting guest lectures, expert lectures, and we expect that all the students will participate in these guest lectures, expertise lecture, or whatever the activities we are conducting, the participation from the student is expected. And any other work related to above the smooth functioning of the academics, as and when the emergency occurs or as and when the cases are upcoming, Then we will take some other measures to conduct the academic activities. Now we all know that from 15th February, government has given the permission for 50% of the attendance. So we are going to prepare a different timetable, and based upon that timetable, you have to come to the college, you have to conduct your practicals or classes, whatever is planned in the timetable, and see that everything is conducted properly. Now these are some of the events which we celebrate, which we carried out during the entire academic year. So we celebrate World Pharmacist Day, that is on 25th September of every year. World Pharmacist Day is celebrated. So we conduct a lot of events, lot of competitions are conducted, rally is carried out, and every year Pharmacy Council of India will give a theme of celebration. So based on that theme, the events will be carried out. Then we have Symphoria, that is one day conference or one day national level workshop or seminar. Every year the topic will be different, and that we generally carry out in the month of January. This year, because of COVID-19, we have not decided to carry it offline, but in month of March, it is planned to carry on online mode. So if you are interested, you can participate in this Symphoria activity. Poster competition is there, guest lectures are arranged, and so the session will be open for all types of students. Then we have Sports Week, cultural activities and annual day in month of January of every year. This year it is not conducted because of the reason you know, but whenever the offline mode will start, college will activity will resume normally, then we'll have these all types of activity. We also have Science Fest and Republic Day celebration in month of January, that is Science Fest 25th and 26th January we will have every year. Then graduation day will be carried out in month of February or March as per the notification of university. This is for the pass out student to collect their degree certificates. A small function will be organized. And industrial visit every year you will conduct. Final year it will be for 3 to 4 days, but up to first, second, third year it will be for 1 day or 2 day industrial visit. So we expect your participation in all these activities. Now this is the second parameter. Now I have covered all the academic guidelines. So what you should, as a revision, what is important is your attendance. See that attendance will not drop. Then your participation in, sorry, participation in different activity. And whatever the problem you have regarding the academics, you can contact your respective class in charges. Sorry. So we have Nisha madam as class in charge of division A or division 1, and we have Varsha madam as class in charge of division B or division 2. So based upon your division, for any academic related query, you can contact your class in charges. Now we will go to the part 2 of our discussion, that is what you should look for. That is when you come to the college as a student, what are the different areas of your interest. So first interest you should be having is to collect knowledge and information. So we have a library with more than 6,000 books for diploma, degree, B.Pharm. All the books are available. So you should utilize the library facility at the most. Second thing is understanding the concepts. So whenever the lectures are conducted, if you have any doubt, you should ask the doubt at the time of lecture, or you can always contact the faculty as per your convenience and get your doubts clarified. So we expect a sort of interaction. We believe in interactive participation or interactive teaching learning process. So doubt clarification, guidance, mentoring, all that is expected when you come to the colleges for your offline education. If you have any questions, you can directly ask the questions to the mentors. Now you will get the mentor. You can get clarification from the mentors, or you can get clarification from the respective faculty on any topic. That may be academic related, that may be non-academic, that may be your personal, or whatever it may be. We are open to all types of clarifications. Reference books, as we have discussed that as you have entered into a professional course, we expect you to read more number of books. That is one method of seeking the knowledge and seeking the information. There are no tutorials, there are no tuitions for pharmacy course. You may be having a habit of attending the tuitions or coaching classes up to standard 12th, but for pharmacy, we don't have any private tuitions, any private coaching classes. All the information what you will get will be only from the faculty, from the classes, and from the books. Whatever the books you are reading, all the information will be available in the books. So don't rely on any information that there are coaching classes, there are private tuitions. It is nothing like that. You, faculty will help you in getting the notes. They will help you in guiding you for the different books. But the ultimate efforts should be done by the students. And so we expect that you should read more number of reference books. Don't rely on very simple books or different the PPTs given by the faculty. It is not sufficient. So you have to put the reading habit as far as the professional courses are considered. Then next, what we expect or what you should look for, that you should prepare your own notes and assignments. The meaning of preparing your own notes is that by referring the different books based on the topic taught in the class, you can make your own notes, so that will help you. At the time of exam, you can go through that notes and you can write the answers of good or better quality. Assignments in the form of writing the journals or whatever the assignment is given by the different faculty, you have to prepare your own assignments. Generally, what we observed is that students will copy the journals of their own friends and they will submit the journals, or they will copy the assignments of their friends and that same will be submitted. That will not give the marks. So you should have the habit of writing your own journal, making your own notes, and submitting your own assignments. And last one is that what you should look for is the respect to the institute, respect to the teachers, and respect to your colleagues or to your friends. Because we are one family, we consider the students as the part of our family. So same we expect from the students, and so that sort of respect is expected for all the stakeholders. So that person may be a teaching or it may be from a non-teaching fraternity. Same type of respect should be given, not only to faculty but to yourself, to your friends and everybody on the campus. We expect in a very harmonious relations. So this is what we expect or what a student should look for when they are coming in the institute or when they are available in the campus. Now we are going to the second point, that what you should not look for, means what we don't expect from the students. First thing, what we don't expect or what you should not look for is missing of classes and practicals. As we have very short time, time is very less, syllabus is very vast, we expect that you should attend all the lectures and you should attend all the practicals. You should complete your journals, assignments, whatever is the task given to you, that task should be completed within the time. Second thing, what we don't expect is the inattentiveness in the classes and laboratory. So we seek active participation, be present in the class, focus on the lecture, ask your doubts, interact with the faculty, try to learn new things, and see that the outcome of the day is there. So just don't attend the lectures or practicals for the attendance purpose or for the sake of attendance. We expect your active participation, and that's what we also look forward. Third thing, what we don't expect from the student is copying the notes, journals of their friends. We want everybody to go in the library, read the books, prepare your own notes, prepare your assignments, search on your own, take the help of the faculty. You can consult your friends, but don't copy the intact journal or intact notes. That will not going to help you in a long run. And last, what we don't expect is the disobeying of the instructions. Lot of instructions will be given to you online, offline. There will be some activities which are time bound, and we expect that all these activities should happen in that time bound frame, and so all the instructions should be followed by the students. Now we are coming to the third part, that is the how to prepare your own notes. First part we discussed was about the academic guidelines. Second part what we discussed was about the what we look for and what we do not look for. And now the third part is how to prepare your own notes. So first you should know that what are the different learning styles. As far as the different psychologists are considered, they have gathered these different, or they have collected these 7 learning styles. Everybody will have different learning style because every human being is a unique person. Everybody learns from their different styles. But if you make a collection of different styles, approximately we will have 7 different learning styles. So which are those 7 different learning styles? First one is visual. By looking to the presentation, by looking to some object, we learn something from that object or from that matter. And so visual learning is very common. We look to the notice board, we look to the blackboard, and we learn something, whatever is written on that board. By reading that, by looking to it, we learn something, and that is called visual or spatial learning, which is a very common learning method. From childhood, from childhood to adult stage, we learn many things only by looking to it. Second thing or second learning style is oral, that is auditory. By listening to some music, sound, we learn many things. So be a good listener. Listen very carefully in the class, listen very carefully in the laboratory, and you can learn many new things only by listening. So develop that skill of listening, because we speak too much and we listen too less. So it should be exactly opposite. Third method is verbal. By speaking, uttering the words in the form of speech and writing, we also learn many new things. Fourth learning style is physical or kinesthetic, by body language, by touching, by the hands, or by different senses what we have. We can learn most of the things by all these physical or kinesthetic methods. Logical is also one of the learning style. We call it as a mathematical. So logic, reasoning, other things, or so social or interpersonal learning is also possible. So I request all of you to mute your audio. Somebody's audio is on, so I request to switch off that audio. It is creating disturbance to other students. Thank you. Last one is solitary or intrapersonal learning style. So either you can work alone or either you can make a group and you can learn many of the things. So adapt all these 7 learning style in your 4 duration of the B.Pharm course, also in post graduation and also for your further study. If you make a mix of all these 7 learning styles, you can grasp the knowledge very fast and very easy. Then how to make your own notes? This is just for your information. I will not say that this is the correct method of making your notes. Everybody will have their own method, but just to make you aware about the basics of making the own notes, you can refer to this slide. First step is doing the basics right. Unless and until you understand or understood the concept, don't prepare the notes. Otherwise, you are laying the wrong foundation. So first, let us make the basics clear, collect the correct information, and then put that correct information as a part of your note. Second step is reading the material in advances. So if you want to make a note, read the material in advance. Read as much as you can from the reliable sources, from the books, and then put that information on the piece of paper as a part of your note. Then third step is review your notes. Whatever you have prepared, whatever you have put on the paper, read it, clarify it, and then finalize it. So unless and until you are assured that this is the correct information what I was looking for, don't finalize the notes. Prepare the most of the notes during the lecture. See, if faculty is delivering the lecture, you can write down the important points and you can memorize that important points as the part of your notes. Because after the lecture, you may forget the important points. So prepare most of the notes at the time of the lecture. Note the main points, that is the highlighted points. Whenever the faculty tells you about the important point as a part of examination or as far as the examination is concerned, so make that points, note down that points during the lecture itself. Put the examples as the part of your notes, so you will remember that point very easy or very easily, or you will not forget that point and you will be able to write the answers in the exam. Clarify the doubts as far as possible. So more clarification you are seeking it, the more basics you are clearing off. And preparing the notes after the lecture, you have to rely on most of the books whenever you are preparing the notes after the lecture. So this is not the complete information which I am sharing with you. This is just a sort of basics. You can modify all these methods. You can adopt your own method for preparing the notes, or you can take the help of faculty, you can take the help of your friends, you can take the help of your mentors, and you can seek more clarification on how to make the own notes. Extension to this is the reviewing the notes, merging the notes, making the outline of the important notes, putting the page numbers to the notes, and the mind mapping. You can see the diagram of the mind mapping, that what can be remembered for longer time. If you want to recall what you remember, I think that all of us will remember the what we have seen, what we have heard, and what we have noted. That will remember for a longer time. So visual, or audio, visual, kinesthetic, sensors, all this will help you to make most of the notes. So whatever you learn, mind or map with your mind so that you will never forget it. It will remember with you for your entire life. So pharmacy is not a subject that you should remember only for the 4 years or 2 years of your postgraduate. It has to be remembered throughout your life, because education will happen only once, but you have to utilize it for your entire life. So academic related information, if you have any doubt, you can message me or you can email me. I have given my number as well as my email ID. For more clarification, you can contact me whenever you have a doubt. We are always there to clear your doubts. So this was from my side. I have elaborated you on academic guidelines, what we expect, what we don't expect from the students, and how to prepare your own notes. So if you have any doubt, the session is open for the clarification. Anybody having any doubt?
Sir, our exam is offline or online mode?
See, your college has just started now. So first we'll finish the syllabus. Till March or April, government will come with the new guidelines. If government says that it should be conducted online, then it will be online. If government says it should be conducted offline because it will happen in April, if the condition improves, then it may be permitted for offline. Then you will have the exams on offline mode. Today I cannot commit on anything, whether it will be online or offline. It will be too early to commit. But based upon the government guidelines, we will communicate to you. Is it clear?
Yes, sir.
Yeah. Any doubt? Any other student having any doubt?
Sir, from where we buy reference books?
See, reference book, there is not a single reference book. For one subject, you may have to refer 5 to 6 books. So instead of purchasing, it is better that you utilize our library, go through the books, and if you find that one book is very important, it is easy to understand, then you can purchase the books from the bookseller. Our librarian has lot of contacts of the booksellers. You can also get the books from that booksellers, or many of the books are also available on online, Amazon or Flipkart. Most of the reference books are available. You can also get from that. But don't purchase now. Come to the college on 15th whenever you are asked. Go to the library, see the books. If it is important, then and then only you purchase. Otherwise, you can get the books from the library. Clear?
Yes, sir.
Yeah. Any other doubt? Anybody is having any doubt? Okay, so I think there are no more clarifications. So thank you everybody for joining the session. Have a great day ahead. So I will request Nisha madam to carry out the session. Okay, so I will request Prashant sir to guide you on the session.
So, good afternoon.
Good afternoon, sir. Good afternoon, sir.
So, I am Prashant Chaturvedi, Assistant Professor at St. John Institute of Pharmacy and Research. And I am looking here for the training and placement in the pharmacy department. So initially, I will go through few slides and then we will do a general discussion. So from the morning, your discussion may be a technical one, but this discussion will not be that much technical. Okay, so it's open for discussion, you can interact. So basically, the importance of training and placement whenever you are selecting a college or choosing a college for your graduation, this is a big concern or a very important parameter which you want, which you are searching for, that how is the training and placement in the institute, one thing. And second thing, what are the efforts which our institute is taking to place their students or to train their students. So initially, I will discuss the facility which is available at our place for this purpose. So the first thing is which we have for you is a CREPT. The full form is Corporate Related Employability for Placement and Training. It's a centrally acting dedicated cell. Centrally acting means it's a one cell which is working for the whole campus. And this cell is working and the placement related activities for the campus. So from pharmacy, I am representing this CREPT center. The second facility which we have at our institute is CSSPD, which is Center for Soft Skill and Personality Development. Again, it's a separate dedicated cell, well equipped with a language lab and a dedicated team of trainers. So this is to develop a good communication skill and soft skill in the students. So there are some in-house faculties and there are some outsourced faculties also, which are conducting some courses on communication, language, soft skills improvement, resume building, and how to prepare for the interviews. So both kind of facilities are available, like in-house faculties are there and we are outsourcing also. So this facility is there for training and placement purpose. The third thing is skill training for employability and placement, which is called STEP. STEP is basically a facility for the certification courses. Actually, what is happening in STEP, that during your 4-year tenure, if you are feeling that there are few subjects which are not being taught in the syllabus, but they are in demand in the market, like nowadays this artificial intelligence or big data analysis or this research or business forecasting. So this kind of things are very much demand in the market, but not in the syllabus. So if students want, they can suggest such kind of courses to the faculty or to me. I will see that what are the options available in the market to whom we can contact, which can conduct such kind of training for you. at the ease of your place, because they will come to the college, because the students are present here in the big number. So the trainer will come to the college. You will not go to their training institute, and we can find out the ways that how it can be conducted smoothly at a very economical things. Then the next thing is a training and placement cell. So this is a dedicated training and placement cell for the pharmacy department. I with Jagan sir, Mr. Jagan, we are taking care of this training and placement activity. In your after the end of your 6th semester, there is a training of 1 month. So we are placing all the students to the different companies, pharmaceutical companies in nearby places or at the Mumbai, where you will go for the 1 month internship. And after completion of your program graduation, of course, you will go for the placement. So we are just finishing your graduation. We used to take a feedback that you want to pursue your higher studies or you want to go for the job. And if it is job, then there are different kind of jobs are available for the pharmacy graduate. Do you want a job in a sales and marketing, or you want in research and development, or you want in quality assurance or regulatory affairs, or in pharmacovigilance? So nowadays, many IT companies are providing the job related to pharma. Like we have a preoccupied perception that TCS, Cognizant, they are the IT companies. But nowadays, this TCS and Cognizant is also recruiting pharmacy graduates for the data analysis purpose, because they are getting a huge amount of the data from the pharmaceutical companies. This data belongs to the clinical trial data, which is difficult to manage manually. So the pharmaceutical companies outsourcing their work to the IT companies, and then IT companies hiring pharmacy graduate and the postgraduate for the data management purpose. So this is a new kind of thing which is coming and emerging, and they are paying also good, and the work culture and other things are also very lucrative. So this is all about summer training, which I have already discussed here. Here I have given few examples that where our students are getting placed. So you can see here, I have named few industries. Of course, there are many more industries because we have an intake of 100. So we have to look for many industries where you all can be placed. So I have given a few names like Cipla, Lupin, Ipca, Mankind, McLeod's, and some nearby companies like B.Pharm, Samruddha, and Neon Pharma, they are in the nearby area. So we are contacting those companies, and we are in very good terms with them because we are placing our students from last many years in those companies. Then the placement purpose. So again, if you have seen the college website, we have updated our placement data of the last up to 18-19. The 19-20 data has not been updated because the COVID situation, the placement is still going on. It is delayed. So the placement is still in process. So I have named a few companies where our students are getting placed regularly, which is one is GSK, which is GlaxoSmithKline, and Lupin, Abbott, Ipca, Teva Pharmaceuticals, Bliss, Neon, Cognizant, TCS are few. But again, this is not the exhaustive list. There are many more companies where we are placing our students. If I will talk about the current year placement, we have already placed our students in companies like Abbott, GSK, in Cognizant, in Lupin, and few students has been placed at Bangalore, one company of the pharmacovigilance. They are placed there. But of course, as of now, they are working as a work from home. When things will normalize, they have to go to the Bangalore. So that kind of portfolio is available to choose. So for the placement purpose, I will say that it's a mutual activity. The things should come from your side also. You do good in your academics, that is a one part, but simultaneously you should improve from your soft skill part also, communication, body language, how you interacting with the external world, how you are making your portfolio strong by participating in many extracurricular activities, so and doing some certification courses. So that will add up to your resume and give weightage to your resume so that you will be placed in a better way. So this is all about the training and placement facilities which are available here. If you have anything to discuss further, you can discuss. It is open for discussion. If anybody has any doubt related to training and placement, career option, job opportunities available after pharmacy, you can discuss. So if you don't have any doubt, we can conclude. Okay. So as
Sir?
yeah.
Sir, when we get our license, pharmacy license?
Pharmacy license only after getting your result of the final year SEM 8. You can apply for the license and it will take 2 to 3 months to get processed, and then you will get it.
Okay, sir.
But that is not a limitation for your job. You will get the placement even before your license. So license has nothing to do with your job. Okay?
Okay.
Okay, fine. So I am handing over the session to Nisha madam. So she will guide you that what will happen next.
All right, so good afternoon, everyone. I know it's already 1:15, so we won't be taking your much time. I think we have already went through this syllabus discussion, a short syllabus discussion in our lecture itself. So I'll just start with my presentation. Those who don't know me, I don't think every one of you will be aware of me. So my name is Nisha Vijayan. I am class in charge of class A, division A, and I am teaching you SUTIX. So I'll be going through the SUTIX syllabus, both for the theory and for the practical. Yeah, I believe everyone can see the slide. So we'll just start with the slide. Okay, so this is the two subject in your semester 1, which is related to the SUTIX. SUTIX 1 theory, which is of 3 hours per week. Remember, it's 3 hours per week. And SUTIX practical, which is 4 hours per week. All right. So now we have already started with the theory of pharmaceutics. We are yet to start with the practical. So I will be elaborating on the practicals which you will be performing rather than the theory. So this is your syllabus, and I believe I have shown you it in the classroom also. So generally the objectives usually consist what you are going to learn in the entire semester. When the semester is done, जब आपका semester पूरा खत्म हो जाएगा, so आपको किस बारे में knowledge रहेगी. और obviously you have to be familiar with the history of profession of pharmacy. Then the basic dosage form. I believe सुबह से आपने dosage form बहुत बार सुना होगा. So I am not going to go into that. Pharmaceutical incompatibilities. Then pharmaceutical calculations, which we are currently doing, and all of you are doing very well in the calculation chapter. So then the way of handling the prescription, which is a very crucial thing. I would say it is a very crucial thing of becoming a pharmacist. If you are not able to handle a prescription, then being a pharmacist is kind of difficult. Then conventional dosage forms, which I had told you is related to the semi-solids, suppositories, which you will be learning through the journey. All right. Now you have different units in your theory syllabus. First unit, which has been started by Shinde sir, it is historical backgrounds, all right, dosage forms, prescription, posology, and as I said in the lecture, posology is something which relates to how much dose एक जन को कितना dose देना है. As I explained to you that your dose will be different, whereas a kid's dose, a 5-year-old child or a 3-year-old child's dose will be very much different. So based on the age and the body weight, we calculate the dosage form. Now, second unit is what we are learning at the moment. That is what I am teaching, and you are all learning, hopefully. Pharmaceutical calculations, all right. Then powders. Powders is also a form of dosage form. If you want a very simple example in our day-to-day life, it is the ORS powder, ENO, all right. Then liquid dosage form is very popular. Syrup, cough syrup. It is generally classified as cough syrup. Okay. Then unit 3 is monophasic liquids, like your ear drops, nasal drops, syrups, elixir. Elixir is something which involves alcohol in it, then your lotions. Now biphasic liquid is something जो दो liquid miscible नहीं होते, like your oil and water. But we have to make a pharmaceutical drug out of both of them. So what we do is that we make it in the form of emulsions and suspensions by using suspending agents. You can see suspending agents and emulsifying agents. So monophasic वो है जो एक दूसरे में घुल मिल जाते हैं, right? They are dissolvable in, they are soluble. But biphasic liquids are the both things which are not so much compatible with each other. So we have to force them to be compatible with each other. Like when people, when teacher makes you sit next to a person you don't like, but by mistake आपको उसके बाजू में बैठना ही, कोई option नहीं. The same way, so the dosage form which results is suspension and emulsion. Now fourth unit is suppositories and pharmaceutical incompatibilities. Suppository is something related to rectal or vaginal drug administration. You will learn about it, okay? And the regarding points which is related to suppository is displacement value, वो सब पता चलेगा, and calculation evaluation of suppositories. Evaluation is a very important term because आपने drug बनाया, you made a drug, you made a medicine, you have to evaluate it like a tablet. The tablet should either be in round in shape, it should be oval in shape, or it should be soft gel capsules. All right, so you have to evaluate it. उसका color बरोबर है, its shape is बरोबर, the thickness is बरोबर. All right, even the bioavailability, मतलब आपने कहा like paracetamol, it helps to decrease the fever. Is it decreasing the fever? Is it decreasing the temperature? So you have to evaluate the formulations which you have made. Now you are not going to be making it in a large scale, but at least you have to understand the evaluation parameters. यहाँ से अगर आप industry गए and they ask you what are the parameters, evaluation parameters for certain dosage form, you cannot just raise your hand and say I don't know. All right, you have to be aware with all the points. Then pharmaceutical incompatibilities. The last one is semi-solid dosage forms. All right, semi-solid dosage form includes everything like paste, even your toothpaste. Yes, your toothpaste is also a pharmaceutical product. Creams, gels, hair gels, then ointments. Okay, now always remember it is ointments. The T is silent. It is not oilments. Do not write oilments in any of your paper. All right. So now we will move on to the practical. Sorry, first I will give you the reference book. So far you are not in the college, so you cannot use the library. So what I have done is that I have referred the Lackman. All right, you can see the 6th reference. Generally it is the easiest book to understand. Then the answer. So these are the 2 books which I have referred and made the PPT presentation, which I will be giving you along with the notes. So just I will also share the syllabus copy. Okay, syllabus copy for all the subjects. I will share it with you. For now, you have to understand that these 2 are the books. Once you come in the college, if you have any doubt, or feel free to refer the books from our library. Now let's move on to the practical. Now practical for your first semester Sutix 1 is divided into 9 parts. Okay, and in total 9 parts has many experiments to be performed. First one is syrup. Now syrup is just like your gulab jamun ka pani. All right, it is sugar and water, but in pharmaceutical industry it has its own reference. If the medicine is bitter, obviously no one likes a bitter medicine. Usually when you administer a bitter medicine to a kid, he is supposed to throw it out. So we add this syrup as a flavoring agent which gives a sweetening taste. Now compound syrup of ferrous phosphate is something which we use as an anthelmintic. Okay, second one is elixir. जैसे मैंने बोला, elixir has like 40% of the elixir. 100% आपका एक bottle में elixir है, so 40% is alcohol in it. Now alcohol has its own advantage. All right, now alcohol of pharmaceutical use, it dissolves the substance also. If insoluble particles are there which is insoluble in water, it might be dissolved in alcohol. Alcohol acts as a preservative itself. So क्या होता है, you don't have to add any extra agent for preservative purpose. All right. A third is linctus. Linctus is like your cough syrup, which is used for the throat complaints. All right. Then solutions. Now the B1, cresol with soap solution, is the general example I can give you. I believe everyone has seen an ad for Lysol. The Cresol with soap solution is a Lysol. Okay, so it is a disinfectant. Obviously, it is not to be consumed. So you will be making that also, because that comes under the pharmaceutical dosage form. I won't say it doesn't come under dosage form. It comes under pharmaceutical forms. All right. Then fifth one is suspension. As I said, suspension is something we use as a biphasic liquid. Okay. The general example which you will understand is calamine lotion and aluminum hydroxide gel, which is also known as Gelusil. Okay. So you all will be making these things. Then we will move on to the sixth, emulsion. Liquid paraffin emulsion. It is a white color emulsion which is generally used on the skin. Okay. For some deficiency of vitamins, it is applied on the skin. Then seventh one is powders and granules. ORS powder. Effervescent granules is like your ENO. When you put it into water, the effervescence comes. It is generally carbon dioxide. And dusting powder is like your Pond's powder. Or there is also itch guard powder. Then divided powders is something we use if जो active ingredient आपका है, it is in very minute quantities. That is the time when we use a divided powders. Now, eighth one is suppository. We will deal with that when we come to it. Then semi-solids, gargles and mouthwash. So these are the 9 classification which you have to perform in the lab. There is no need to worry. All of them are very easy if you understand the procedure ones, which I will be explaining when you come to the college for the practical. It is very easy, but you have to pay attention to all these stuffs. Okay. So that is that for your syllabus. I hope everyone is clear with that.
Yes, ma'am.
Yes, ma'am.
So,
Yes, ma'am.
so next, we will start with the next introduction to the subject, which will be given by Ms. Sapnali Shinde. You are already familiar with that
Ma'am?
because she, yes.
Ma'am, can you again go through the books?
Books. Please write down the name itself. Lachman. Lachmann. Lachman. And the second book is Ansel. Actually, in our field, we usually recognize the book by the author's name. All right. Ansel, Ansel. So next will be HAB session. Enjoy. Thank you.
Okay, good afternoon, students. I am Ms. Swapnali Shinde, subject in charge of Human Anatomy and Physiology 1. So I am here to discuss the syllabus of HAP, that is Human Anatomy and Physiology 1. So it is very exciting to learn and gain in-depth knowledge of human body as well as different systems of human body. So in detail, we'll learn about different systems, and this HAP, that is Human Anatomy and Physiology, this is the subject, basic subject, and it is related to the pharmacology. So we can say this is one base of your pharmacology. So first, we'll strengthen the knowledge about human body in first year, and then in advanced year you will learn about different drugs action on these systems. Okay, so first we'll start with see, you have this theory subject as well as the practical human anatomy and physiology. You have 3 hours per week for this and 1 for tutorial. So total credit points, that is 4 for theory, and for practicals you have 4 hours per week. So 2 credit points for practical. So upon completion of this course, the objective of this subject, that you should be able to explain the various structure as well as the functions of organs of the human body as well as different systems, Then various homeostatic mechanisms are there. So in detail about that, maintenance of body temperature. Then you have identification part, so identification of different tissues, then the organs of the systems, and as well as you have to learn about different special senses and about the nervous system. And at last you have working pattern of this different organs of each system. So you will come to know about everything about anatomy and physiology part. So here in human anatomy and physiology 1, you have total 5 units. The first unit, that is introduction to human body, cellular level of organization and tissue level of organization. So the very first topic, that is introduction to human body. In this you will learn about the normal terminologies that are used in this HAP. So you have what is anatomy or what is physiology. Apart from this, you have different structural organization of all body systems, as well as the basic life process. So this is there, the basic part is there in 1.1. Then you have cellular level of organization and the tissue level of organization. So in cellular level, you will learn about the plasma membrane, different types of cells, then the cell junctions, then you have cell communication and cell division. So this is about the cellular level. Apart from this, you have different pathways regarding this cellular level. So okay, so different pathways are there. So if you have any drug and it will cross the plasma membrane, so based on that you have this pathways. So that will cover in this point, that is cellular level of organization. Next you have tissue level of organization. So in tissue level of organization, you will learn about the anatomy and physiology of all types of tissue. So you have 4 types of tissue, that is epithelial, then muscular, nervous and the connective tissue. So this is about the unit 1. So it is very basic one. So you will learn about different 12 systems of your body in detail. Second unit, that is about integumentary system, skeletal system and the joints. So in integumentary system, you have structure and functions of skin. And apart from this, you have different accessory organs which are related to skin. So in detail, you will learn about skin. Then the second point from second unit, that is about skeletal system. So it will cover the whole part, that is the skeleton. So you have different types of bones, then the features and functions of all type of bones. So you have identification of axial bones as well as identification of appendicular system. So in skeletal system will cover the organization of this skeletal system. Apart from this, you have joints. So in joints, the classification of this joints, structure and functional classification of joints, and in detail again there is identification of different joints. So this is about the second unit, which will cover your skeleton. Third unit, that is very important, the blood and the body fluids, and you have lymphatic system also, which will cover in third part. So about blood, so this is the very important point, very important fluid. So you have composition of blood, different functions of blood, and you will learn about hemopoiesis, so production of RBCs, then WBCs. Then you have blood group system, so ABO system is there, RH factor is there, the disorders related to this blood which are there, that also will learn in short. And along with this, you have different blood clotting mechanisms, so that will cover in this third unit. Second important point from this third unit, that is lymphatic system. So lymphatic system, that means structural and functions of lymph nodes, then spleen. So basically you will learn about the immunity system over here. So lymphatic system, it will cover the lymphatic organs, lymphatic tissues and lymphatic circulation. This is about third unit, so which will cover your blood as well as the lymphatic system. Now the fourth one, that is the peripheral nervous system and special senses. So from nervous system, you have only one part, that is peripheral nervous system. So again, in peripheral nervous system, the anatomy and physiology of this nervous system, you have two divisions, that is sympathetic and parasympathetic nervous system. So in detail about sympathetic and parasympathetic nervous system, and along with this you have other kind of nerves, that is spinal and cranial one. So in detail we'll learn about that in PNS. Then in unit 4 you have special senses. So the structure and functions of all your senses, like you have eye, ear and nose, and along with this you will learn about disorders which are related to this all senses. So this covers your 4th unit. The last unit, that is the unit 5, cardiovascular system. So already we started with this cardiovascular system. So here you have anatomy of heart. So in that the location, different chambers of heart, as well as different walls which are present, then the blood flow through this heart, and apart from this you have blood circulation via pulmonary and systemic circulation. Then you have cardiac cycle and cardiac output. So you will learn about the conducting system of heart, which will cover the SA node and AV node. And apart from this, in detail you will learn about blood pressure and the ECG. So that is electrocardiogram. And for cardiovascular system you have disorders. So we'll cover arrhythmia in this session, that is the cardiovascular system. So this is about your theory part. Next we'll cover see the practical one. So in human anatomy and physiology you have this 15 experiments. The very first, that is study of compound microscope. So how to operate this microscope or how to handle this microscope, that will learn in this experiment. Then second part, we can divide this practical syllabus into 3 parts, that is the introduction part, then the microscopic study, and determination of different hemocytometry. So second part, which will cover the microscopic study of all types of tissue. So you have epithelial, connective, muscular and nervous tissue. So that will learn in this second and third practical. Then you have about the skeleton system. So that is the identification part. So identification of axial as well as the appendicular bones. Next you will determine the WBC count, RBC count, then the bleeding time, clotting time, then you have blood group determination, then erythrocyte sedimentation rate, and heart rate and pulse rate. So you will count this everything related to hemocytometry of your own blood. So we'll see that in lab in detail. And at last you have recording of blood pressure. So basically this practical, you have the microscopic study, identification part, and determination of different parameters which are there in hemocytometry. So this is about the theory as well as the practical of human anatomy and physiology. Now see the reference books. For HAP, for theory, the very first book, that is the Tortura, that is Principle of Anatomy and Physiology by Tortura. This is the whole and soul of this HAP, and the in detail again you have this in pharmacology also. So this is very important book. Apart from this you have Anatomy and Physiology in Health and Illness by Wilson. So Ross and Wilson is there. Then you have the Essential of Medical Physiology. So these 3-4 books are very important. Same you can refer for practicals also. See over here, these are the reference books for practical. So we'll share, if you have the soft copy, we'll share with you all. And again you have notes for all these points, as well as we'll share the PPTs also. So you will get enough material to learn this human anatomy and physiology. Okay, so this is about the human anatomy and physiology. So we'll end this discussion. I hope this is clear.
Yes, ma'am.
Okay, fine.
Yes, ma'am.
Okay, so next Varsha ma'am will take the session.
डायरेक्टली स्टार्ट करूँ मैम, डायरेक्टली स्टार्ट करूँ ना.
Good afternoon, everyone. Am I audible to everyone?
Yes, miss.
Yes, ma'am.
Slides are visible?
Yes, ma'am.
Good afternoon. Myself, Varsha Godke. I'm the class in charge of B division. You heard my name several times. Now, this is the introduction, and I'm also the class subject in charge of the pharmaceutical analysis. Now, directly we'll start the introduction part. I already discussed this syllabus copy in our lecture. Pharmaceutical analysis theory and the practical. Here the theory which contain a 3 hours in a week, one tutorial, and which having the 4 credit points. And the pharmaceutical analysis practical which is having for the 4 hours per week. Now let's discuss the syllabus. Now the scope of this subject. This course deals with the fundamental of the analytical chemistry and the principle of the electrochemical analysis of a drug. The analysis itself explains that it is having the some chemical, electrochemical methods to find out something, okay, or to find out the testing or to perform the testing on the drugs and the chemical which we are using to perform different practicals. Now the objective, upon the completion of the course, students shall be able to understand the principle of the volumetric and the electrochemical analysis, carried out the various volumetric and the electrochemical titrations, and the develop analytical skill. This is the purpose of this subject. Now the theory part, the pharmaceutical analysis, what exactly it is, the definition and scope of it, different techniques of analysis, which techniques exactly which we are going to use to perform the testing of the chemicals and the pharmaceuticals. Then methods of expressing the concentration, primary and the secondary standard, the preparation and the standardization of the various molar and the normal solutions. Here we express the concentration of the solution in the normality, molarity. Now this, what exactly the preparation and the standardization of this particular oxalic acid, sodium hydroxide, hydrochloric acid, sodium thiosulfate, sulfuric acid, potassium permanganate, and steric ammonium sulfate, which we are going to under this unit 1. Now the next is a B part of the unit 1 is the error, the source of error, types of error, methods of minimizing the error. When we are going to performing some methods, now that time some error will happen. So what exactly the source of this, then what are the types of that, and the methods of minimizing, what exactly the method we are going to apply to minimizing the error, then the accuracy, precision, and the significant figure. Then pharmacopoeia, the source of impurity and the medicinal agent, limit test, all are, and we are going to study under the unit 1. Now unit 2 is, there are the types of the titration. Acid base titration totally depend on the pH of your solution. The theories of acid base indicator, classification of acid base titration, what are the theories involved in the different titration of the strong acid, strong base, weak acid, weak base, and the neutralization curve. Then non-aqueous titration, the solvent, acidometry. Non-aqueous titration, the solution which are not soluble in water. For that solution we are going to perform the non-aqueous titration. The solvents, acidometry, alkalimetry titration, and the estimation of the sodium benzoate and the ephedrine HCl is the example for this non-aqueous titration. Now unit 3 contain 3 different types of titration, that is a precipitation titration, complexometric titration, and the gravimetric titration. In precipitation titration there are 3 methods: Morse method, Volhard method, Fajans method, and example of this precipitation titration is the estimation of sodium chloride. In complexometric titration, what is a precipitation titration? During the titration it forms one insoluble substance which we call it as a precipitant, PPT. Okay. In complexometric titration it forms the complex with the ions which we are present in the solution. Now the classification of the complexometric titration: metal ion indicator, masking and demasking reagent, and the example of this complexometric titration is the estimation of magnesium sulfate and the calcium glucurate. Now the next is a gravimetry. So the principle and the introduction part of the gravimetry, there are several steps which we are involved to perform the gravimetry analysis. The purity of the precipitate, co-precipitation, post-precipitation, estimation of the barium sulfate is the example for this gravimetry analysis. Now the next is a diazotization titration, the basic principle, method and the application of diazotization titration. Unit 4 contain a redox titration. Now you all know the concept of the oxidation and the reduction. So this redox titration is totally dependent on the concept of oxidation and reduction. There are several types which include in this redox titration is serimetry, iodometry, bromatometry, dichlorometry, titration with the potassium iodide. Now your next unit 5 contain the electrochemical methods of analysis. In this electrochemical method means what? It needed an electric supply or the electric instrument to perform the analysis on the particular chemicals. In this unit, the chapters are conductometry, potentiometry, polarography. Conductometry, the induction, then the introduction, then the conductivity cell, conductometric titration and its application. In potentiometry, there are electrodes, 2 electrodes that we have to use to perform the potentiometry is the reference electrode and the indicator electrode. The types of reference electrode is the standard hydrogen silver, then silver chloride electrode and calomel electrode. And indicator electrode is a metal electrode and the glass electrode, and also the methods to determine the end point of potentiometric titration and application. Now the last part is the polarography, the principles, Elkovic equation, construction and the working of the dropping mercury electrode and the rotating platinum electrode and its application. All this we are going to discuss in this semester in a theory part. Now goes on the practicals. Now your first practical is the limit test. Limit test why we perform to calculate the purity of a solutions. Okay. Now the limit test of the chloride, sulfate, iron, arsenic. These 4 limit tests we are going to perform in our practical lab. Next is the preparation and the standardization of sodium hydroxide, sulfuric acid, sodium thiosulfate, potassium permanganate, ferric ammonium sulfate. How to prepare the solution for this and how to standard and how to calculate the normality. This study which we are going to study under this second part. Third is the assay of following compound along with the standardization of a titrant. Now how much amount of the particular substance is present and what is the percent purity of that particular compound, like the ammonium chloride by the acid base titration, ferrous sulfate by serimetry. All these are the types of the redox titration. Copper sulfate by iodometry, calcium gluconate by complexometry, hydrogen peroxide by permagnetometry, sodium benzoate by non-aqueous titration, sodium chloride by precipitation titration. The titration processes which we are going to discuss in a theory part, also we have to perform in the practical one by one. Determination of the normality by the electroanalytical method. Here in a theoretically we can determine the normality by using the N1V1 formula, but in a practical we can also determine the normality by using the electroanalytical methods like the conductometry titration of the strong acid against a strong base, conductometry titration of the strong acid and the weak acid against strong base, potentiometric titration of the strong acid against strong base. Okay, now these all are the reference book for the pharmaceutical analysis. I also put this reference book into the your Google Classroom for your reference. Okay, now the bucket and the Stanley which is the group which is the book I'm going to refer to prepare my PPTs. Then Vogel is also there, Bentley and Drive, then John Skinley and the Indian Pharmacopoeia. These all are the books we can refer to study the pharmaceutical analysis. Now this is all about your pharmaceutical analysis subject, introduction part of the theory and the practicals. If you have any doubt, you can ask.
Miss, can you show the book?
I'll share it on the our Google Classroom. Wait. See, you can take the picture of this.
Thank you, ma'am.
Okay. Now this is all about your pharmaceutical analysis introduction part. Now I'm going to hand over the session to the Mr. Tanveer sir.
Hello, good afternoon everyone. I hope I'm audible clearly. Let me know.
Yes, sir.
Yes, sir.
Sir.
Good afternoon.
Yes,
We are
sir.
going to have our session of induction on this pharmaceutical inorganic chemistry. I hope you probably know me very well. My name is Tanvir Singh Rajput. I'm working as an assistant professor in Department of Pharmaceutical Chemistry, and we will start our session by using this slide as a reference from the syllabus itself, which contains two subjects from the PIC. One is for theory aspects, another one is from the practical. Theory consists of 3 hours, while practical has a 4 hours. Credit point is in front of you. As I started in my first session itself, I just decoded this concept of pharmaceutical inorganic chemistry into the two parts, that is inorganic chemistry and addition to it is a pharmaceutical. You probably knowing the term inorganic chemistry very well as a student of science, that this chemistry is different from that organic chemistry in the sense that we cannot have that backbone of carbon hydrogen skeleton. That means we are free from that skeleton of carbon hydrogen, and any compound or substance which does not contain carbon hydrogen backbone will be classified under the inorganic chemistry, and we are going to study it. But we are not going to study every inorganic chemistry substance. We have been limited ourselves to the concept called as a pharmaceutical, and pharmaceuticals are the substances or drugs which are there to treat, cure, mitigate, diagnose various forms of diseases, and we will limit the inorganic chemistry only those substances which will be helpful for you to cure, treat, diagnose the diseases. Regarding the monograph, which is the integral part of this inorganic chemistry, that is pharmaceutical inorganic chemistry, I said I just make an analogy with your Aadhaar card. That monograph is something like that, an identity card which consists of all the information related with that inorganic drugs and the pharmaceuticals. Like in case of Aadhaar card, you will have your personal information as well as the biometric information. But in case of monograph, which are encoded in your so-called Indian pharmacopoeia, British pharmacopoeia, and all the pharmacopoeias, consist of a variety of information starting from the molecular weight to the molecular formula, then solubility to the assays and identification test, uses, precautions, and all that. So you are going to have a brief and even in-depth discussion with respect to the monographs of all the inorganic drugs and pharmaceuticals. What is the objective or outcome of this PIC course is to know the sources of impurities and methods to determine the impurities. It is an integral part of pharmaceutical industry because you cannot produce the drug with 100% purity. It's presumed to be that there are certain ways by which impurities may get incorporated. And that's why if you know the sources of impurities, then you can have a very much chance, a good chance to eliminate the impurities also. And apart from this, you are going to understand the medicinal and pharmaceutical importance of inorganic compounds. In the theory part, in the unit first itself, impurities, you are going to aware with the various forms of impurities and limit test itself. But before, you must have completed with the pharmacopoeia in its history. As I said in my earlier lecture also, that pharmacopoeia will be considered as a Bible of pharmacy. If you don't get anything related to the pharmacy, you just visit any pharmacopoeia, maybe Indian, maybe British, maybe US pharmacopoeia, and you will get the information regarding this. Just like the holy books which we refer, Vedas and Puranas and Bibles, these pharmacopoeias are there to help every pharmacist and every pharmaceutical scientist to gain some knowledge regarding various drugs and their monographs. So in the first chapter, we will have the discussion on the impurities, like the limit test for the chloride, sulfates, iron, arsenic, lead, and heavy metals. Apart from this, there are modified limit tests for chloride and sulfate. The same has its replica in the practical itself. So it will be very easy for you to comprehend it because you are going to handle the same procedure, principle in the practical itself.
Most importantly, the syllabus consists of asterisk mark on the particular compounds. These compounds are very much considered to be important from the point of view of pharmaceutical inorganic chemistry, and you need to be go into depth of that asterisk marks compound from methods of preparation point of view, assay of that particular drug, properties, medicinal uses of inorganic compounds belonging to those classes. In the unit second itself, and you must be probably knowing the different subjects, this PIC has much of the overlap with respect to the analysis and physics also. You will be probably knowing to presume that this will be there, like the adjustment of tonicity, isotonicity was a part of the pharmaceutics itself. So in the acids, buffers, bases and buffers, we will come to know what is buffer equations and buffer capacity, what is the relationship between them, what are the pharmaceutical applications of buffers, and most importantly the calculation and the methods of adjusting the isotonicity becomes an integral part of this unit too. Apart from this, major extra intracellular electrolytes, that means what are the physiological functions of various electrolytes, what happens if they are in the deficiency, what are the replacement therapies for them, and monographs of respective electrolytes like sodium chloride, potassium chloride, calcium gluconate, ORS, physiological acid-base balance. Then there is a dental products. It consists of various excipients like, or rather various modes of dental products like dentifrices, desensitizing agents, what are the role of fluoride in the dental products, and the monographs of calcium carbonate, sodium chloride, zinc, eugenol cement. Antimicrobials, mechanism, but apart from mechanism, you are going to have a little bit of familiarity with the concept called the bactericidal, bacteriostatic, what is the difference between them, how antimicrobials exactly act, what is its mechanism, what are its classification, and apart from this, the antimicrobial agents and their monographs like potassium permanganate, boric acid, hydrogen peroxide, chlorinated lime, iodine, and its preparations. Unit 3 consists of gastrointestinal agents, and it consists of acidifiers, antacids, and cathartics. That means the GI agents, and working on the GI agents, what its impact on the GI diseases, like acidifiers are there, antacids are there, which works on the hyperacidities or even the ulcerative colitis. So exact monograph of aluminum chloride, dilute HCl, then ideal properties of antacids, combinations of antacid, and including the monograph for sodium bicarbonate, aluminum hydroxide gel, magnesium hydroxide mixture, and in cathartics, you will going to have a monograph of magnesium sulfate, sodium orthophosphate, kaolin, and bentonite. And the unit 4, miscellaneous compounds, which consist of a variety of categories like expectorants, emetics, hematinics, poison and antidote, and astringents. What exactly the term called the expectorants, emetics, and various other categories, and what are their so-called monographs associated with that particular category, maybe potassium iodide, ammonium chloride, copper sulfate, sodium potassium tartarate. We will have a discussion on it. Lastly, the radiopharmaceuticals, which we have started. What is radioactivity? What is half-life? How you are going to measure the radioactivity by using various units of radioactivity? How the properties of alpha, beta, gamma radiations are different. Then there are several pharmaceutical applications of radiopharmaceuticals, including sodium iodide 131. What are the storage conditions and precautions and handling strategies with respect to the radiopharmaceuticals? We will have a discussion on it. This summarizes the part of the theory. Then there are certain practicals associated with the PIC, which has its overlap with respect to the theory part, and that becomes very easy for you to understand. Like limit test is there in the first chapter itself. So limit test of chlorides and sulfates, then ions are there, then heavy metals, arsenic. Identification tests are there for magnesium hydroxide, ferrous sulfate, sodium bicarbonate, calcium gluconate, copper sulfate. Certain tests for purities like swelling index in case of bentonite. We will have the first practicals of this also. Neutralizing capacity of aluminum hydroxide gel, determination of potassium iodide and iodine in the potassium iodide. Apart from this, there is a preparation of inorganic pharmaceuticals, specifically ferrous sulfate, has to be performed at the practical desk. These are the recommended books for the theory and practical as per the university syllabus. Beckett and Stanley, one of the very good book for understanding the practical aspects of inorganic chemistry. Vogel's book, another one for understanding the theoretical aspects. Gundu Rao, as per the PIC syllabus, hence most preferred. And you can also go for Anand and Chatwal, and remaining monographs will be dealt in the Indian Pharmacopoeia. I hope you have cleared with the PIC now. If you have any questions, you can raise it over here.
Can you show the slide of book?
Slide up. Okay. Beckett and Stanley for the practical aspects of PIC. Ogles, Gundu Rao, Anand Chatwal will be helpful for theoretical aspects, and Indian Pharmacopoeia is there to help you to analyze and understand the various monographs of inorganic compound. Is it clear now?
Yes, sir.
Yes, sir.
Yes, sir.
Any other question? So I presume that you have cleared with the PIC. We will meet in the theory sessions. Till then, take care. I will hand over the session to Nisha madam.
All right, so I don't know about you, I definitely need a break. We all need a break. So we'll take a break of 10-15 minutes and be back by 2:10. All right. Now, when I say we'll take a break of 10-15 minutes, be back by 2:10, it means the next session is about your examination. How you should write, what you should do, what not you should do, how is your marks calculated and all. So be back by 2:10. All right. So you guys can just log out now. The session will be online only. If you want to keep it like that, keep it like that. The session will be like this. All right. So you can join back later. Thank you. 2:10. Do not remember 2:10.
Means if a student absent for a reason, so after that he can get a chance to give a re-exam. Then? Okay, thank you, ma'am.
मैम सेशनल एग्जाम कब होगा? सेमेस्टर के पहले होगा।
Yes, sir.
No, sir.
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir. Yes, sir. सर सर अगर एनएसएस में पार्ट लेना है तो उसके लिए भी कोई प्राइज मनी है क्या? अगर सर हम लोग को पार्ट लेना है एनएसएस में तो उसके लिए भी कोई मीन्स पैसा देना पड़ेगा क्या? ओके, थैंक यू। ओके, थैंक यू सर। ओके, ओके सर, थैंक यू।
सर एक्स्ट्रा अटेंडेंस के लिए 120 आवर्स रुकना पड़ेगा क्या? ओके, थैंक यू सर।
सर वो जो 7 डेज का रेसिडेंशियल कैंप रहता है वो हर दिन लेके जाते हैं या फिर एक ही बार लेके जाएंगे? 7 दिनों के लिए उधर रहना पड़ेगा?
चलेगा सर।
सर लेकिन पास में ही है ना धानु, किधर दूर है? रोज भी तो जा सकते हैं। ओके
सर
सर।
आ जाऊंगा।
ओके सर। मत सर फिर दोनों में आना पड़ेगा? ओके सर। ओके। ओके सर। यस सर। थैंक थैंक यू सर। थैंक यू सर। Good afternoon, ma'am.
Which day? Okay, it was good. It is good.
Good,
Yeah,
miss.
ma'am, it was.
Yes, ma'am.
Ma'am, what is your name?
Wow. मैम ज़्यादा हो गया था मीन्स ओवरलोड हो गया है. But it was amazing, ma'am.
Yes, miss.
Yes,
Yes,
miss.
ma'am.
Yes, miss.
Yes. Yes, miss.
Someone who guides us. No,
Got
but
it.
No, ma'am. Yes, ma'am.
Yes,
yes.
ma'am.
Yes, ma'am.
Yes, ma'am.
Yes,
Yes,
ma'am.
ma'am. Yes, ma'am.
Yes, miss.
Yes, ma'am.
Yes, ma'am.
Yes, miss.
Yes, ma'am.
Okay,
Okay,
miss.
ma'am.
Yes.
Yes, miss.
Yes, miss. Yes,
Yes,
miss.
ma'am.
Yes, ma'am. So, yes, Tanvir Singh Rajput, sir. Tanvir Singh Rajput, sir. Yes.
Ma'am, Tanvi Singh Rajput sir is my mentor.
Ma'am, my mentor is Navnath Hatwat. Ma'am, my mentor is Nitin Sen, sir.
My mentor is Ramesh Busal, sir.
Tanveer Singh Rajput, sir.
Prakash Khandagle, sir.
Sahaya Nadar.
तीन तीन 17 है मैम. Yes, ma'am. Prashant Dalvi, sir.
My mentor is Nitin Sen.
Yes, ma'am. Varsha Godke, ma'am.
प्रशांत चतुर्वेदी सर.
वर्षा घोड़के मैम.
जगन्नाथर सर.
नितीन सेन सर.
नितीन सेन सर.
तनवीर सिंह राजपूत सर.
तन्वीर सिंह राजपूत सर.
प्रशांत दलू.
नितिन सेन सर.
अविनाश दर्शा सर.
तनवीर सिंह राजपूत सर.
प्रशांत शिंदे सर.
स्वप्नाली शिंदे.
ईशा मैम.
नितीन सेन सर. नॉर्मा रबीलो सर. सॉरी मैम.
बेला वर्गीस. अविनाश बचा सर.
स्वप्नाली शिंदे मैम. नितीन सर.
सुदर्शन सर.
प्रवीण सर. ईशा मैम.
गोविंद आसने सर.
प्रवीण पवार सर. सुदर्शन सर. ज्ञानेश्वर सर.
सहाय मॅम.
वृषाली मॅम.
प्रशांत गवली सर.
भरत सर.
तन्वीर सर.
गोविंद सर.
प्रकाश सर.
निशा मॅम.
प्रवीण सर.
नितीन सेन सर. नयनाथ सर.
मीता मॅम.
नवनाथ सर.
प्रवीण सर.
स्वप्नाली शिंदे मॅम. स्वप्नाली मॅम.
सुदर्शन सर.
तन्वीर सिंग सर.
प्रवीण पवार सर.
नितीन सेन सर.
प्रवीण सर.
मीता मॅम.
Yes, ma'am.
Yes,
Yes,
ma'am.
yes,
Yes, ma'am.
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
And what is the procedure to change mentor? Okay, okay. Okay, ma'am. Okay. Yes, ma'am.
Yes,
Ma'am,
ma'am.
your
Yes,
voice is
ma'am.
not
Yes,
clear.
teacher.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am. Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Yes, ma'am.
No, ma'am.
No,
No,
ma'am.
ma'am.
No,
No,
miss.
ma'am.
No
No,
doubt.
miss.
Yes, ma'am.
Yes, miss.
Yes,
Yes,
ma'am.
ma'am.
Yes.
Yes, miss.
Yes,
Yes,
ma'am.
miss.
Yes, ma'am.
Yes, ma'am.
No, ma'am.
No, ma'am.
No, miss.
No, ma'am.
No,
No,
ma'am.
ma'am.
No, miss.
Yes.
Yes, ma'am.
Yes.
Yes, miss.
Yes, miss. Yes, miss.
Yes,
Yes,
ma'am.
miss.
Yes, ma'am. We will make sure. Okay, miss.
Okay,
Yes,
ma'am.
Okay,
teacher.
miss.
Yes.
Yes,
Yes,
miss.
Yes,
ma'am.
miss.
Yes,
Yes, ma'am.
Yes,
ma'am.
miss. Yes, ma'am. Yes, miss. Yes, miss. Yes, miss. Yes, miss.
Yes, ma'am.
Yes.
Yes,
Yes,
ma'am.
yes, miss.
Yes, ma'am. No, ma'am.
No, miss.
Yes,
Yes,
ma'am.
miss.
Yes, ma'am.
Okay.
Thank you, miss.
Thank you, ma'am. Yes, miss.
Okay, miss.
Okay, ma'am.
Thank you, miss.
Thank you,
Ma'am.
ma'am.
Thank you,
Thank you,
ma'am. Thank you, ma'am.
miss.
Thank you, ma'am.
Thank you, ma'am.
No, miss.
मैम हमारी हालत भी वही है.
मैम फ्रेशर्स पार्टी रहेगा क्या? नहीं मैम, कुछ नहीं. मैम वो रिक्वायरमेंट लिस्ट वापस से दिखाओ ना. ओके मैम. Thank you, ma'am.
Thank you.
Thank you, ma'am.
miss.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am. Ma'am.
Thank you, ma'am.
Thank you, ma'am.